All right, welcome everyone. Welcome to the Eastern U.S. Floodwater Summit, Solutions for Floodwater Mosquito Control. My name is Leanne Lake and I am with Valent Biosciences. I'm a technical development specialist. And welcome everyone to the summit. And thank you for taking the time to spend with us today and learn from all of our amazing speakers that we have and all the time you're taking out of your schedule today to be with us. And we have an amazing group of speakers today with us. I'm going to go ahead and share my screen with you so you can see my presentation real quick. Can you guys see my presentation now?
No, we can't.
All right, let me just share my screen here one more time if I can find that. Not sure why I don't have that option right now. Banu, I cannot, I share screen does come up for me. So, oh, here we go. All right. I think we're in business now.
Yeah, we can see it.
All right. Let me just go ahead and put this in full view for you guys. All right. Welcome. Sorry about that. Welcome to the Floodwater Summit. Like I said, we have some amazing presentations today and a full schedule. So we're going to keep it pretty tight today and move along really quickly. I do have a few household items that I need to go through quickly before we get started. So I'm going to do that really quickly. First of all of your microphones are already off out there, so you don't need to worry about that. We cannot hear you. So there are a few ways that you can communicate with us. There is a Q&A portal at the bottom of your screen, which you can use to ask questions to the speakers or to us and other things like that. There's also a chat option, and that chat option, we're going to try to use that specifically for some pop-up quizzes that we have for CEUs and other things like that. So reserve the Q&A for all your questions and things like that, and the chat we'll use for just those CEU requests that we have coming through for the pop-up quizzes. If you need some additional assistance, this program has an entire BBC team in the background helping us out. So we have Caroline in the background who'll be working the chat and working the Q&A, and you may hear from her throughout the presentations here and there helping me out. We also have Banu in the background who's going to be helping us with just the Zoom platform and some other things. So I am going to give him the table real quick to make sure there's any other items he wants to cover in the household housekeeping items. Banu, please take it away for a second.
Yeah, thanks, Leanne. Good morning and welcome everyone for the Floodwater Summit Eastern webinar. Just a couple of things for the chat. Leanne already mentioned, please use the chat window for only important questions, mainly because we want to have that pop-up quiz available for everyone. And if there are too many people chatting, the pop-up quiz will actually keep going higher and higher. You have to scroll to find it. So Q&A, please use the Q&A button on that Zoom platform that you see. And regardless of whether a speaker is speaking at that time and you have a question on some other speaker is speaking, you have a question for another speaker, whatever it is, just put it on the Q&A and it'll actually maintain all the questions until we answer them. So even if we don't get time in between a couple of speakers, we will be able to come back to yours as and when we get time to get back to those questions. So type away your questions on the Q&A portal so that we know that those are the questions that need to be answered. We won't be monitoring the chat window for questions specifically. So that's all I had. Leanne, go ahead.
Okay. All right, great. We got one more person in the background working with us. Chris is in the background. He is helping with people getting in and out of the platform and things like that. So if some people are having issues getting in or other email issues and stuff like that, he's with us to help. I did drop his email here if you need him and you or share it with a coworker who can't get in. All right, so that's the team behind the backgrounds who'll be helping us out. And I'm very thankful for all of them for helping us in this adventure. All right.
Can you hear me by chance, Leanne?
Barry, I can hear you. Thank you. You got it. So
Okay.
welcome.
Good to go.
All right, then we're going to go in regular order. So, all right, Ceu Basics. This is what a lot of you are really interested in. So I want to touch on this just briefly so you understand how this is going to work. CEUs are distributed after the event. So if you attend the entire event, which you must, then you will get an email from me that says claim your CEUs here. All right. So when you click that link that we provide, it'll take you to a list of all the states that have approved CEUs. You're going to click on your state and it will take you to the form that you need to fill out for your particular state. And it will ask you the information such as license number, name, all that information that your state requires for CEUs. And that is your claim form. In addition to that information, there's a short quiz that you must fill out and pass with 70% or higher to get your CEUs. And it is connected to that email that I send you after the event. All right. Now there is also going to be a secret code word that I'm going to give towards the end of the summit. And you are going to need to enter that code word on that CEU claim form. Okay. And this is another way you have no idea when it's coming for me to make sure that you're tuned in and that you're paying attention. All right. So some states, you're going to receive a CEU certificate after this event is over. In some cases, they actually have to require original signatures from me. So please just give me a few days to get through this whole process, sign those, and actually mail them to you. We do have a few days to finish all the paperwork for all the states. So for everyone, please give us a little bit of time, maybe a week or two to get everything submitted to your state. And then you should see those CEU forms coming to you around that time. All right. Again, if for some reason you do have a question, you're not getting a form, there's an issue, please reach out to Carolina or myself, and we will take care of you individually to get you through that process. All right. In some cases, there are some additional application forms for renewal, and those instructions will be in your CEU link. But if you need additional help with those forms, please again reach out to us and we'll help you specifically with whatever questions that you have to get that paperwork filed and filled out correctly. All right. There are a few states that I want to point out a couple of items. If you are in the state of California, Utah, Colorado, and the last state is blocked for me, I'm sorry, Oregon, that's right. You are required every hour to take a pop-up quiz. And this will be, the links are going to be dropped in the chat box for you to click on the link and take that pop-up quiz. It's only going to be there for a few minutes, so you need to be paying attention for them to pop up and make sure you fill all of them out and submit. Some of you have already received an email about this, but there is an option to get a text message to you and receive those pop-up quizzes on your phone via text message. All right, if you want that option, my team is going to drop a link in the chat box now for you to do that option and you'll receive them like that. State of Virginia, you do have one additional requirement at the end of this that is attached to your CEU form that you'll get from me. And that is the legal presentation that you must go through and complete to claim your CEUs. All right. One very important thing to everyone. It is in violation of every state agency that we have gotten approval for, CEUs, for participants to lie or intentionally mislead us about their identity and or the participation in this event. So in the forms that you fill out, there will be a statement to check off to saying exactly that I am who I am and I did participate. All right. I think that covers it mostly for the CEUs. Carolina or Banu, is there any other last minute items before we get started?
There are a couple of questions that popped up on the Q&A portal, so you might want to answer those. So one was, are there credits for New Jersey?
No, there are not. So we apologize. There is a requirement there that we could not get around regarding IDs and identification process. So sorry, we did work very hard on trying to get that accomplished, but we just could not. So.
Okay,
Yeah.
Second question is, hello, your summit has an impressive list of presentations. In lieu of CEU credits, will attendees receive a certificate of attendance?
Not automatically. So if you would like that certificate, just because there is 600 plus attendees today. If you need a certificate of attendance, we do have them. Reach out to Carolina and I and say, hey, please send me one and we'll get it right out to you. But we're not going to drop it to all 600 just because we didn't want to confuse you with your actual CEU forms that we are sending you that you need to submit to the state. So we just didn't want to flood you with too many documents. But if you do need that document, we do have them prepared. Send me or Carolina an email and we'll send it right out to you.
Just to remind attendees from Colorado, Utah, California, and Oregon about pop-up quizzes that will come up in the chat box. So just stay tuned.
Yep.
You will need them.
There was a third question, and Carolina already answered that one, but I would still like to bring that up. Someone was actually asking I cannot see my video. And the reason is you cannot see your video. You can only see the presentations and the video of the panelists. The reason is this is actually a webinar, not like a regular Zoom meeting. This helps us because there's a lot of attendees here. So if we turned on everybody's camera, it'll be a lot of cameras. So we want to focus on the speakers. We have some good presentations going on and the speakers are giving good talks. So we want to focus on them so we can get this content across. So that's the reason you are not seeing your camera.
All right. Awesome. We're going to move on now and get started. If you have more questions, just drop them in there. We'll reply privately or to the group as we need as we get started with our presentations. So I'm going to go ahead and introduce our first speaker. We have Dr. Barry Alto, who is an associate professor of Arbovirology at the Florida Medical Entomology Lab here in Florida. And he is going to be presenting Ecology of Floodwater Mosquitoes. So Dr. Alto, you're on. You can share your screen and take it away.
Okay, thank you, Leanne. Is my volume all right?
Yeah, I can hear you.
Okay, and I should be sharing my screen right now. Can you see it?
Nope, right now we still see you.
Try to share my screen.
Down towards the bottom, there's a share screen with an arrow up.
And show.
The green color button.
Yep, then click your
Share show, okay.
share.
Screen one.
Yep.
You gotta choose the PowerPoint or either desktop 1 or 2, whichever one screen that you have the PowerPoint on. But I just, there you go. We're seeing,
Okay.
yeah.
Now you're seeing it. Now I'll put it into slide mode.
Yeah.
Slide. Okay.
There we go.
Gary, Barry, actually you have the wrong screen selected, so we can only see your notes. It's not the right screen, so it's presenting to the second screen.
Okay, let me redo that again. Sorry about that. Let's pick the other screen.
So you have to first stop sharing. And then choose the whole power. You can either choose 1 or 2 desktops, or you can actually choose the PowerPoint file itself. That way you can actually show the PowerPoint regardless of where.
Okay, so, okay, stop sharing. All right.
Hit share screen again.
And now I pick the other screen, not 1, but 2.
Yeah, that'll work too. Yeah, now go to, yep, yeah. And then hit the PowerPoint into presentation mode, it'll show up here automatically. There you go.
Right. Okay, well, thank you for the introduction and the opportunity to make a talk here at this workshop. So, larval habitats of mosquitoes, with over 3,500 species of habitats and climate, these are what largely determine the distribution of mosquito species, as well as biological interactions among species. Larval requirements may be highly specific, as is in the case for those who specialize on breathing apparatuses through plants, or they might be quite general as well. Here are some examples of running water occupied by some anopheles and Culex, transient floodwater, which we'll focus on today. Here's a wood pool, 80s stimulants, permanent water, there's a freshwater swamp, and container systems that happens to be a natural container systems. So there's been numerous attempts to provide categories of distinct habitats for mosquitoes, especially in reference to the aquatic habitat. And for this presentation and sake of simplicity, I'll refer to 4 categories, the ones that I just mentioned here. In each of these habitats, I have mentioned an example of a mosquito species that may be found there. So temporary flooded locations. These include flooded pastures, for example, Seraphra species, fresh flood water. Here's a ditch on the side of the road, as well as tidal floodwater, Aedes sollicitans. These agricultural lands are enormous mosquito producers, including Aedes, Seraphra, and Culex being mainly these floodwater type mosquitoes. Transient water sources or flooded waters, snow pools, and ditches are used for the breeding grounds for these species. And these eggs that they lay on the soil can withstand desiccation, sometimes for years, such as in Aedes and Seraphra. Their life cycle is in tune with this type of adaptation, is in the sense that they require alternated periods of wet and dry. So this is a key feature of their biology. Now, opportunistic Culex, like Culex nigripulpus, might be able to utilize the habitat for a single generation during an extended flooded period, but they're not typically the ones that you'd think be, laying their eggs or egg rafts on soil. So floodwater mosquitoes, they are in the genera Aedes, Ocleritatis, and Seraphra, and these are 3 genera that are the most important pest species. They're highly nuisance mosquitoes. They bite humans, livestock, and pets. So they certainly affect us and the things that we take care of. And in many instances, there's large populations in the spring and early summer associated with rainfall events. Eggs are laid on the surface soil at the edge of the standing pool of water, in this case, that are left from rains or floods. Often woodland pools, roadside ditches, or low areas along creeks, rivers collect this water. The eggs hatch when flooded by runoff or from heavy rains or flood water. So let's go into a few examples of them and talk a little bit about their biology and ecology. So Seraphra, Columbii, and ciliata, these are associated with sun-exposed ephemeral water, pooled water in agricultural lands and distributed in grassy landscapes. They're also found with other Aedes. There's huge abundances of Seraphra columbii following periods of high rain, and it is a common nuisance to livestock, rabbits, and humans. The peak abundance is usually early summer, June to July, depending on the availability of larval habitats. And you can see on the right side of the screen an example of each of the mosquito adults and their distribution shown in gray in the United States. Fairly large mosquitoes, and when you get bit by them, you usually know it. Okay, the dark rice field mosquito, Seraphra columbii. These eggs are deposited in humid soil by gravid females. And notice on these eggs that there's spine-like tubercles, and they allow it to adhere to the substrate. And this is an adaptation they have to laying in this type of moist environment. The larvae, and you can see a few pictures of them here, are white, sometimes brown, they're long. They have 4 clusters of CT located at the top of the head, and you can see that in the bottom left picture. Each of these CT have 4 branches. The anal papillae are long, translucent, and taper to a point, and that's in the far right lower picture. Seraphra columbii is a possible vector of canine heartworm, although 25 species of mosquitoes contribute to transmission throughout the same range. It also may be a vector of epidemic strains of Venezuelan equine encephalitis virus. Now, Seraphra ciliata, there's really no evidence as a bridge vector for viruses, but it has been detected to be positive in nature for a number of different viruses shown here. So Seraphra ciliata are primarily mammalian blood feeders, so they will feed on our ruminants, including our livestock, armadillos, raccoons, and rabbits, and they readily feed on humans and can be annoying, especially in high abundances. Aedes taeniorhynchus and Aedes sollicitans, these are salt marsh mosquitoes. The black salt marsh mosquito on the left and the eastern salt mosquito on the right. They are associated with large emergences and they're very aggressive biters. They're not considered a primary vector of major disease concerns, but they may be involved in triple E, V, and dog heartworm. They're found along the Atlantic and California coasts, breeding in salt marshes, as well as other parts of the tropical Americas. They occupy floodwater, hog wallows, rain pools, roadside puddles, and other temporary bodies of waters. These, like others, are laid in moist soil that are sheltered high enough above the water line to be affected only by sufficient flooding. And it's that aspect of the biology which is really important, and it shows that when you know some of the basic biology of the mosquito, you can exploit that biology to better control them. For example, using an integrated mosquito management approach, larviciding and source reduction, impounding and ditching. So when water levels are high, they're not going to lay their, or when they're standing water, they're not going to lay their eggs there. And this is a way of making impoundments and protecting certain areas, especially those that are located near residential areas. And in the picture here, you can see a person digging a drainage ditch for source reduction. Aedes vexans, they lay their eggs individually on moist soil above the water line in a variety of aquatic habitats. And after a short period of drying, the eggs must subsequently be flooded with water to hatch. Now, during periods of drought, eggs can remain dormant for years. So this is quite a special feature of this mosquito. It is multivoltine, so it is able to produce several generations each season. And typically, adults live 3 to 6 weeks, sometimes longer, depending on the environment, and allows them to lay several broods of eggs. And it is considered to be primarily a biting nuisance mosquito. Culex nigerpulpus, the distribution in the United States is shown in the upper right-hand map here, shown in gray. Some larvae and a blood-fed adult here. These eggs are reported in most types of aquatic habitats, but best described as a floodwater Culex. So it's a little bit different than some of these other ones. One of the most important disease vectors in Florida, and it is a disease vector of St. Louis encephalitis virus and West Nile virus. It has a subtropical distribution. It's found in the US, the Caribbean, Mexico, Central America, and parts of North South America. And the larvae develop in rich organic mixtures in shallow flooded ditches. Culex nigerpulpus is involved in epidemic transmission of SLE and West Nile virus, and possibly it may have some involvement in Eastern equine encephalitis virus. So what's unusual about the biology and behavior of this species that makes it a vector? Well, it has, first of all, it's opportunistic in its choice of hosts, in part due to host abundance and feeding during crepuscular periods. So what makes it a primary vector is that it is attributable to this egg dumping behavior, which gets synchronized with some other attributes of its life. So females prefer to lay eggs in freshly flooded ditches along droughts during, and I should say, in long droughts during the summer limit these sites. Okay, so if there's not many sites, they're going to withhold their eggs. So what happens is that you have many mosquitoes, sometimes up to 90% of the population of the mosquitoes are holding gravid eggs. And then after a rainfall event where it breaks the drought and sites become available, it synchronizes oviposition, seeking hosts, and blood feeding in a cycle that is also synchronized with the rainfall. If the period of the drought is 10 to 14 days, then the other feature that makes it the primary vector is during that period of time, it approximates, depending on the temperature, the extrinsic incubation period of some of these viruses that it transmits. And so you have a mixture of the abiotic conditions combining with some of the innate behaviors of the mosquito and interacting with a pathogen that under some circumstances can create a perfect storm. And that is it for my talk. And I think I have some time if there's questions.
Yeah, awesome. Thank you. Let me pull up the question thing here. We do have one for you. Is there a niche partitioning between Aedes sollicitans and Aedes taeniorhynchus? Or do they compete? If so, how is I think that's supposed to be who's the more successful competitor?
Yeah. That's a good question. And I don't think that there's been a lot of studies that have looked at competition, whether it be in the adult stage or the immature stages for these mosquitoes. Clearly, they do have some differences in their larval habitat. But beyond that, I don't know a lot of the details that I've read from the literature. I'm more of a container-dwelling mosquito person myself. Sorry, I can't be of more help.
No worries. We have a couple more questions coming in, so let me just scroll down. Is there a study of how long salt marsh eggs can lie dormant?
Yes, there have been studies, and they can lay dormant probably for longer than people would think, certainly days, weeks, and months.
Okay. All right, we do have one more question. Can solicitans survive in fresh water?
To some extent, they are able to tolerate some fresh water.
Okay. Awesome. I think that is all of our questions. So thank you very much, Dr. Alto. And
Sure.
You can stop sharing your screen and we'll go ahead and move on to the next presentation.
Okay, thank you.
All right. Next on our list is Mr. Elmer Gray, a research professor at the Department of Entomology at the University of Georgia. And today he's going to talk about larvicide active ingredients and their roles in integrated mosquito management. So whenever you're ready, take it over.
Hey, Leanne, thank you very much. It's a great pleasure to be here. I appreciate being invited to participate. The integrated pest management approach has been a part of mosquito control long before it was a recognized practice. As early as the early 1800s, there was an area by Savannah, Georgia that switched from wet rice culture to dry rice culture and used local tax monies to do it. So this is kind of an unrecognized mosquito control event. The earliest ditching of the New Jersey salt marshes and the open marsh water management practices that take place today are other good examples of an integrated pest management approach. I grew up on the salt marshes of New Jersey and Ocean County, very familiar with this area and the ditching of the marshes and the effectiveness of the salt marsh mosquito and floodwater abatement there. Before pesticides, this is the way they did mosquito control. So today we're returning to a more integrated approach, and we're going to hear a lot about the different, some of the approaches used for this. The there's many reasons to return to an integrated approach and use an integrated approach. The efficiency and the effectiveness of the control, the sustainability, and public perception. I think we all know that the public's probably going to have more input into our mosquito control practices moving forward than they have in the past. Integrated mosquito management is a kind of a broad topic that I have a little bit different approach to than some people. I serve as the public health extension specialist here in Georgia, and we have many very small programs where there's limited resources and the populace only wants certain aspects of mosquito control done. So while that may not be a comprehensive program, we do have comprehensive programs. Chatham County down in Savannah, as many people are aware, is a great program, uses all facets. But some areas want to just use education, and this involves educating the public as to what they can do around their homes. And I also include educating the workers. We've got, almost 500 people here today. Hopefully everyone will take up, a couple tidbits of information they can take back to their programs. Workers become more confident in their knowledge, and you can deal with the public better. And the more confidence you have when you're dealing with the public, the better it represents the program, the better the public thinks of our programs. Source reduction would be the next step. Permanently eliminate sites, improve drainage and habitat manipulation, the marsh management and flooding practices that occur in the lagoons in Florida, but community cleanups as well. So source reduction is and we can do that without even knowing what mosquitoes are there, because we all know that a roadside ditch that holds water is a problem. Surveillance would be the next step. What do we have and where do they occur? This helps focus our efforts. As with all pest control, it's obviously best to know what species you're dealing with. For mosquito control, it's more important because it tells us where to look as far as the larval habitats. Larviciding would be the next step. We target the habitats that can't be eliminated. Larviciding requires effort and resources. I think people sometimes overlook this. Those of us who have been involved in larviciding efforts know that it's a lot of work, requires boots on the ground and resources. And the final step would be adulticiding. Adulticiding is highly effective. Communication becomes essential. We need to be aware of pollinator concerns, and insecticide resistance is obviously a very important aspect moving forward here. And we do have a cooperative extension circular 1154 that goes into more detail on these. Larviciding has many advantages. The larvae are concentrated in a confined or defined area, particularly compared to the emerging adults. The larvae are essentially immobile, and they're not getting away for a few days, and they're accessible. The CIA approach is something that William Horsfall was a proponent of and talked about in his 1985 50th anniversary presentation at the AMCA meeting. So this goes back a ways. I'll add to that the advantages of larviciding is we typically think that the larvae are susceptible. Obviously there's some resistance occurs, but in general, I think the larvae are more susceptible than some of the adult populations. It's proactive. Obviously we're targeting the larval stage before the adults emerge, and it's more acceptable to the public, has reduced risk to pollinators. Choosing a larvicide is a really important decision, and there's going to be, I think we're going to have some good presentations about this moving forward. Obviously, there's a wide variety of formulations. We have 4 types of active ingredients for very different modes of action. And ideally, the formulation and active ingredient is chosen based on the type of habitat and the mosquito population is present. And that's really what is the fine tune to which product is used, which formulation and active ingredient is the habitat and the stage of the population, how much time you have to get them before they get off and things like this. The more experience and knowledge that the operator or program has, the more targeted and efficient the larvicide application will be. The larvicide active ingredients, we basically have 4 groups. There are insect growth regulators, which are absorbed through the integument of the larval stage. S-methoprene is the most common and has been a long time standard. The Insecticide Resistance Action Committee, or IRAC classification, is a 7A. This describes the mode of action of the larvicide. Pyriproxyfen is also an insect growth regulator that's approved probably for storm drains. I don't think there's a floodwater formulation, so we're not going to talk about that anymore. Just want to touch base on it. The microbial products, these must be ingested, the proteins that are produced by the bacteria, Bacillus thuringiensis, subspecies israeliensis, and Bacillus sphericus. Lincilli bacillus is a new name. I think we've been through the nomenclature changes with mosquitoes, so this is just something to be aware of. I'm going to stick with the Bacillus sphericus moving forward. Most people are still most familiar with that. Surface oils produce a physical barrier. Today, they're highly refined mineral oils, and there's no IRAC classification because it's a physical control agent. Spinosad is effective by contact or ingestion, and this is a biological neurotoxin with the IRAC 5 classification. So we've got 3 different classifications in the physical barrier. So for larvicides, that's a pretty wide range of modes of action, and it's probably a little bit better, a little bit more advantageous than our adulticiding options. The insect growth regulator S-methoprene. This is a natural juvenile hormone that was first identified in 1967. It's involved in the regulation of the physiological processes, particularly metamorphosis, and research quickly followed, developed the first larvicide around 1975 for mosquitoes. It's used in a wide range of insecticides today. My first experience was with hornfly control on livestock. It inhibits an ecdysone from initiating the molting processes. It's particularly disruptive to adult emergence, and malformed adults may be observed on the water surface of treated areas. The pupal stage does not feed, as we're all aware, so they will eventually deplete the body stores of essential nutrients and starve. So it primarily affects the pupal to adult transition stage, and it's effective against all mosquito species. Just an example of how it works, normal mosquito development, it would have high juvenile hormone levels in the early instar larvae. They decline as they age and mature and approach the pupal stage. When you treat a habitat with the juvenile hormone analog methoprene, you maintain these high levels of juvenile hormone in the environment, and it just disturbs the physiological processes of the pupal stage primarily. So again, it's absorbed through the cuticle and by ingestion. The concentration must be higher in the larval environment than circulating in the larval body for disruption to occur. It's used for the wide range of applications. Larvae and pupae don't die immediately, so this leaves them in the food web. This has some advantages. Efficacy evaluations can be challenging because the larvae and pupae don't die. So that's something that operationally technicians need to realize, that you can have a very effective methoprene treatment ongoing and the larvae are still there. So that's something people need to be aware of. And it's a long time standard. There's been many improvements in the formulations. There's formulations available from several of our producers now. Microbial-based products, Bacillus thuringiensis, subspecies israeliensis, or BTI, is probably the most common. Again, the Bacillus sphericus is also available. Both of these materials are very common today. BTI probably having a more prominent role in targeting floodwater mosquitoes. Bacillus sphericus was more commonly recognized as a material used to target Culex mosquitoes, particularly for the reduction of West Nile virus transmission, but the other uses are expanding. It's very effective against the Seraphra mosquitoes that Barry just talked about, and also Aedes vexans. So it definitely has uses in the floodwater realm. And there are combined formulations available, both sphericus and israeliensis, that will provide a wider range of treatment options in a wider treatment zone. Bacillus thuringiensis israeliensis is a bacterium that occurs naturally in soils and aquatic environments worldwide. BTI was discovered in a drying pool in a riverbed in the Negev Desert of Israel in 1976, was quickly produced for commercial products. Thuringiensis had long been recognized for having insecticidal properties. There's other BT products for lepidopteran, forestry products, but the discovery of BTI brought it into the public health arena. There are many serotypes and strains of the BTI bacteria and the different producers use different strains for their actual final products. So that's something you'll see sometimes on the label. The active ingredient of BTI-based formulations are delta endotoxins that are produced at the time of sporulation. The endotoxins are composed of 4 proteins that are activated by the proteolytic gut enzymes in the alkaline gut environment of the Nematosser and Diptera larvae. All right, a lot right there. Nematosser and Diptera larvae are your mosquitoes, black flies, and Chironoma midges. They have a very high pH in their midgut, which is unusual for most animals and organisms. And they have the enzymes associated with that highly alkaline gut are effective against these proteins, cleave them and activate them. Just an example of that, the image there, the midgut of a mosquito larvae. The formulation is protoxins are ingested. They're activated by the enzymes, break them down. The polypeptide fractions, or which are proteins, bind to the cells. They form pores or holes in the lining of the gut cells. An osmotic imbalance occurs. The cells swell, they lyse, the larvae die. This can happen rather rapidly when they're exposed to high concentrations. BTI must be ingested to be effective. Larval feeding is absolutely critical. And the larvae seem to be very susceptible to these proteins in this product. It kills the larvae very quickly that are exposed to high concentrations, slower for those larvae exposed to lower concentrations. And this is something that's operationally, people need to be aware of that, just because low populations killed with, treated with high concentrations may die very quickly, but larvae exposed to lower concentrations still probably will experience mortality. It might just take a longer time. Appears to be the amount of protein they ingest, how rapidly that midgut is disrupted. As I mentioned, there are 4 proteins associated with larvicidal activity, and there seems to be some synergistic effects of these proteins. They're cleaved by the enzymes. You often see about a 25 kilodalton protein. Consequently, there seems to be a reduced risk of resistance with BTI. There's multiple proteins involved. Seems it's a very complicated process and very specific environment where it occurs. There are formulations of BTI that are approved by the Organic Material Review Institute, or OMRI, and that's a very good thing. Obviously, some of the formulations are very clean and can be used in organic production areas. Their BTI has no mammalian toxicity, and the non-target impacts are very minimal. Bacillus Sphericus, another bacterium that occurs naturally in soils and aquatic environments worldwide. It was first isolated in 1964 from dead Culex incidens larvae in California rock pools. There are several serotypes with larvicidal activity, but it has a narrower spectrum of activity, as there are only 2 proteins involved with the toxic effects of it. Has a similar mode of action to BTI, but that's kind of a general statement. It affects different cell structures than BTI, but it is ingested. The toxins are attached to a living spore, and the coating of the spore seems to aid in the residual aspects of the Bacillus Sphericus larvicides. There are also is some recycling of toxicity associated with Sphericus. The greater the larval population in initial treatment, the greater the residual effect. So what's going on there, the larvae are actually ingesting spores, and these spores will germinate and produce bacteria in the larval cadaver to produce a recycling event with the Bacillus Sphericus to extend the residual control up to around 28 days. It is also approved by the National Organic Program. Some of the formulations are, so it also has a very clean profile as far as environmental and organic production areas. Just a quick example of the mode of action. The spores in associated protoxin are ingested. Feeding ceases, the spore coat dissolves, releasing the protoxins. The protoxins, or the proteins, are cleaved by enzymes, activated, and bind to the gut wall, creating pores or holes in the gut wall. The cells lyse, The larvae die, but some recycling will occur because, as you recall, the spore is still there, and the cadaver environment of the cadaver is very conducive to bacterial growth. Surface oils, petroleum oils were the first chemicals used in efforts to control mosquito larvae. Thankfully, we have progressed to a more refined mineral oil-based product today. These are the only pupicides, so that's something to take out of this right here. It's an extremely important aspect. If you've got habitats that have large pupal concentrations, you've got limited options of what you're going to do. And also, they're coming soon. Adult mosquitoes are coming soon. So surface oils, the mineral oil-based products can be used to get these. The effectiveness of these oils are limited to mosquito larvae and pupae that breathe air at the water surface. So your Coquetidia, the mosquitoes that will insert their air siphon into the roots of plants are not going to be affected. Lower rates will minimize non-target impacts. Higher rates are required for polluted water in areas with significant vegetation. This material has got to spread across the surface of these areas, so application will be important as well. It's probably the least studied larvicide. I thought that was interesting. I was looking at some of the Florida information, and John Beadler stated that, and the mode of action is related to the effect of the product on the surface of the water, and there may be some toxic impacts as well. So it's interesting that the oldest material is maybe the least well understood. Suffocation of both the larvae and pupae does occur. This mode of action is completely different from the other larvicides, so it's very difficult to envision how resistance would occur to this material. Coverage is very important, and mortality can be rapid. Spinosad is a biological neurotoxin. It's one of our newest active ingredients. Has a kind of an interesting background story as well. A vacationing scientist took a soil sample from a drum that was used to make rum in the Caribbean. From that sample, a new species of bacteria was identified, Saccharopolyspora spinosa, and this translates to spiny sugar. So during the fermentation of the bacteria, the two most prominent and active compounds in the fermentation broth are spinosad A and spinosad D, hence the term spinosad. This material alters the function of the nicotinic and GABA-gated ion channels, causing rapid excitation of the insect nervous system, leading to involuntary muscle contractions, tremors, paralysis, and death. So this is, kind of your traditional neurotoxin. You got the synapse there and the acetylcholine effects. So it's more of a traditional material and different from our other active ingredients. This neurotoxin is specific to invertebrates, and it is classified as a reduced risk pesticide by the EPA. It has also been approved for use in organic productions areas by OMRI, so it also has a pretty clean environmental and label approach. It breaks down rapidly in sunlight and in soils, and the microbes are going to break down spiny sugars, as you would imagine. It's effective against all larval instars. Mortality most rapid when ingested, but some contact toxicity, and it is a unique active ingredient whose use is growing and is certainly an excellent part of a rotation. So our larviciding options, we have many different formulations that are available. There are combinations of active ingredients that provide more application flexibility, a wider treatment window. That would be the BTI and methoprene combinations, or BTI and Sphericus combinations. It's been going on for a long time. The combination formulations have probably been more refined now in more recent years with formulation technology. Matching the optimal formulation and active ingredient will maximize the efficiency and effectiveness of a larvicide application. That's really something I took out of this as I prepared for this presentation. When I was talking to many of our industry representatives, I highly suggest that we discuss the strengths and weaknesses of formulations with your commercial representatives and other control specialists. Use your resources. To any directors out there, you know, as we return to trade shows after this pandemic, I strongly encourage the support of operational people participating in these meetings and developing a network that is so valuable. There's so much information out there. We can't learn it all ourselves, but our commercial representatives and our control specialists from adjoining counties and states can provide information and improve our programs tremendously. So use your resources. Networking is really important. Product rotation remains an important aspect. We've got a lot of different very good products here. Integrated mosquito management has never been more important. Using all aspects of it: education, source reduction, surveillance, larviciding, and adulticiding. Larviciding can be highly effective in preventing all types of mosquito populations, whether it be floodwater or containers. A lot of work been done in this area in recent years. Larval surveillance and knowledge of the physical parameters of the larval habitat is critical to choosing the proper product and optimizing the timing of the application. You know, that's what we have all these different formulations for, because there's whether you have single brood, whether you have extended release, multiple broods, the timing, late instar, just a lot of different aspects that the various formulations can help us address, and the expertise of our commercial reps can help us choose which one. And of course, being cognizant of non-target impacts, I want to stress that we're always concerned about what else we're affecting. You know, in summary, the wide variety of floodwater habitats can all be satisfactorily addressed by one or more of the active ingredients or combinations that are available. We have 4 very different modes of action: the insect growth regulator methoprene, the microbials and their stomach actions, and being Thuringiensis, Israeliensis, and Bacillus Sphericus. The surface oils, highly refined mineral oils, again, our only pupicides, and the biological neurotoxin spinosad provides a very different mode of action from the other larvicides. Effective surveillance of the target population, site specificity, and cost will often dictate options. I am not so naive to think that cost does not come into play. I do want to stress that there are other costs involved, whether it be susceptibility or environmental impacts. So it's not just the dollar and cents of the product, but all the costs involved. Susceptibility is a big thing, and product rotation. So cost averaging over 2 or 3 seasons might be a better way to approach the cost aspect moving forward. But all the active ingredients can and should play an important role in floodwater mosquito management programs moving forward. Lastly, I'd like to put a plug in for the Georgia Mosquito Control Association. We're going to meet in October, and we want you folks to come join us. Thank you.
Awesome. Thank you, Elmer. We do have quite a few questions and a couple of minutes to go ahead and answer those. So we'll go right through them here. First one: how long does it take for the insect growth regulator to absorb into the larvae's body and take effect?
I can't answer that, specifically. The biggest impact is in the later instar larvae, as I showed that the juvenile hormone levels decline as they age. Temperature is probably going to play a big role in that as well. The warmer it is, the more rapidly it probably can be absorbed. But that's a little bit out of my realm of expertise.
All right. Well,
as many of these may be.
No, we'll go ahead and move on to the next one. Is there a mechanism of resistance to methoprene in mosquito larvae?
There has been examples of some resistance being documented, but there are many areas that have used methoprene for a very long time that do not have resistance. So that's kind of there seems to be some conflicting data on that. Chatham County, I talked to Bobby Mullis, and they've used methoprene for a very long time on their floodwater spoilage areas with great success. So the proper application, I think, is really critical to that.
Yeah, and speaks to what you mentioned about product rotation and the rest of that program of why that stays working well for them. So excellent. Next question. Spheritex works in containers? ? Did you hear that one?
No, you broke up on me.
Okay, sorry. The question was, does Spheritex work in containers?
Bacillus Sphericus, yes, that would probably work very well in containers.
I'm sorry, Spheritex is what they actually mentioned, which I think is more of a brand name.
Not familiar with Spheritex.
Okay. All right. Well, we're going to go ahead and move on. We do have quite a few other questions in here for you later on, but we're not going to get to them right now. So we'll get them in the panel discussion. All right. I do want to shout out real quick. Now is the time for a pop-up quiz for those states that we mentioned before. Again, California, Colorado, Utah, or Oregon. These are absolutely required. And you'll see in your chat a link to take that quiz. And another friendly reminder, there is the text option on your phone, which is also a link to that option in the chat if you want those things. So make sure you take your quiz before we move, move on. All right, next presenter, we have Dr. Tara Tiemann, who has recorded her presentation for us. And she is an associate professor of biological sciences, at the University of the Pacific. And she's going to be presenting on Pyrethroid resistance in Culex tarsalis in the regions of North America. So Bono, if you go ahead and get that presentation.
Okay, so I put a little gray box in the upper left-hand corner. If you want to move the video up there, then I think you'll be able to see all of my slides. Today, I am talking about a project looking at pyrethroid resistance in Culex tarsalis. And this project was primarily completed by two of my graduate students, Emiko de la Vega and Bonnie Ryan. Before I get started, my vector control possible without them.
All right, looks like we're having a little bit of technical difficulties. So
Yeah, I'm gonna fix that. Hold on.
All right, hang on with us one quick second, and I'll plug that quiz one more time. Make sure everybody has that. And I did want to address a couple other things that have been popping up in the Q&A while I have a moment. Those that are requesting the presentations will be available on the VBC website after the event is over. So we'll make sure that you have access to all these presentations. And of course, they are the panelists are going to stay in for the panel discussion, and I'm sure many of them will allow you to reach out to them if you have further questions that we didn't get to today, things like that. All right, Banu, how are we doing there?
Okay, so I put a little gray box in the upper left-hand corner. If you want to move the video up there, then I think you'll be able to see all of my slides. Today, I am talking about a project looking at pyrethroid resistance in Culex tarsalis. And this project was primarily completed by two of my graduate students, Emiko de la Vega and Bonnie Ryan. Before I get started, I want to thank my vector control collaborators. So projects like this are definitely not possible without them. San Joaquin, Saciolo, Placer, and Lake County, all in California, and then the Salt Lake City district in Utah. I worked with them to develop the project. They helped with collecting mosquitoes. For this project, a lot of the bottle bioassays were done at the districts, and they also provided funding. In addition to the funding from the districts, this project is also funded by PACVEC and the University of the Pacific Graduate Program. So I'm going to be talking about Culex tarsalis, probably not news to anyone here. This is an important vector of West Nile virus and other arboviruses in the Western US. Historically, this mosquito is a rural mosquito, though now it is often found in residential areas as well. Because of where it's found, it faces insecticide pressure from agricultural spraying as well as vector control, and until recently, few reports of insecticide resistance had been reported. Now, I will say that over the last few years, that has changed. There have been more and more reports of resistance in tarsalis. A couple of goals for this project. The first one was to determine the prevalence of resistance to specifically pyrethroids in select study sites across Northern California. Then after establishing that there was resistance, we wanted to look at the mechanism. So what's happening in the mosquitoes that can lead to this resistance. We looked both at enzymatic resistance using enzyme assays that happen in microplates. Sometimes they're called microplate assays, and also looking for target site mutations. So to think about the resistance mechanisms, it's important to know how the insecticide works. So here's what is likely a review for you for how pyrethroids work. They target the nervous system of the mosquito. So first, that star there represents where the action happens. The pyrethroid would bind to voltage-gated sodium channels. This binding actually holds those channels open. This increases the influx of sodium ions into the neuron. That increase of sodium increases the number of action potentials, which increases the amount of acetylcholine that is released from the cell. That acetylcholine goes to other neurons and muscles to overexcite them. So basically with this overexcitation, the mosquito uses all of its energy sending action potentials and contracting muscles, and that leads to the mosquito's death. So mechanisms of resistance that combat or that prevent pyrethroids from working. The first one that I mentioned, and I'll say a little bit more about here, are detoxifying enzymes. So this is when mosquitoes actually produce more enzyme that can break down or inhibit the insecticides. This diagram is a little complicated and it shows information for all types of pesticides, or at least for many types of pesticides, but I want you to just to focus on pyrethroids since that's what I'm focused on here. And the takeaway is that the enzymes known as esterases, oxidases, and GSTs have all been shown to lead to resistance for pyrethroids. The second type of resistance mechanism that we looked at are target site mutations. Target site mutations are actually a mutation in the DNA itself. This mutation in the DNA leads to a change in the amino acid and the protein that keeps the insecticide from binding to its target site. For pyrethroids, the mutation of interest is something called KDR, which stands for knockdown resistance. This mutation prevents the pyrethroids from binding to the voltage-gated sodium channels. And there are two common mutations. There have been others discovered, but these are kind of the starting point when looking for resistance. And that is a change from the amino acid leucine to phenylalanine, or from leucine to serine at one place in the gene that codes for the voltage-gated sodium channel. So let me dive into these goals. So first, looking at determining the prevalence of resistance. And for this, we did CDC bottle bioassays. This work happened in the summer of 2018. First, mixed age females were collected using CO2 traps. We had about 3 to 5 populations per district, and there are those California districts that helped me out greatly with this project. For the bottle bioassays themselves, we had kind of 4 categories of bottles. So the control that didn't have any chemical, right, acetone only, that acetone evaporates, leaving just the clean bottle. We had PBO only, PBO being a synergist that inhibits oxidases, one of those enzyme groups that I talked about. Permethrin only, and then permethrin plus PBO. For each of those groups, we ran these in triplicate, so 3 bottles, and about 20 mosquitoes in each bottle. Every time the bottle bioassays were run, we ran a susceptible colony so that we could make sure that the assay was working like we wanted to, along with the wild-caught population. And in most cases, we were able to do 2 biological replicates. That is, we were able to repeat it twice for the field site. After the mosquitoes were in the bottle, we recorded mortality every 15 minutes for 2 hours. Here is a graph that shows the type of data that we get from the bottle bioassays. You perhaps have seen these before. On the y-axis, we have mortality. On the x-axis, we have time. So again, with those 15-minute checks to see whether or not mosquitoes had been knocked down or were dead. The green lines here are for our susceptible colony. You can see that they died early on in the experiment, which is what we want, what we expect. They died in about 15 to 30 minutes. The orange lines here are for the wild population, in this case, Empire from San Joaquin County. And if we look at the permethrin only bottle for this particular population, we can see that mortality neared 100%, but it didn't quite reach it. However, when PBO was added, mortality happened much faster. So in this case, mortality reached 100% at 45 minutes. An example from another population, this is Winters in Yolo County. Again, we have our susceptible colony in green. Now, this is a different colony. The summer we did this, it was actually quite difficult to get colony mosquitoes. A lot of districts, their colonies were down that summer. So we used whatever colony we could find. It's not ideal, but it's what we could make happen for that summer. You can see this KNWR colony took a little longer to die, but still everything had died by 60 minutes and most of the mosquitoes by 45 minutes. Now, looking at the wild population, only 8.6% of the mosquitoes died by the end of the 2-hour point. So quite a lot of resistance in this population. When we added PBO, you can see mortality increases, but in this case, it still never reached 100% by the end of the 2 hours. Now, for the sake of time today and for the sake of everybody's sanity, I'm not going to show you 15 more of these, but we do have the data for 15 other sites. Here they are summarized in this table. So on the very left, you have the district in California. Then we have the column that shows the specific site. These are percent mortalities. So if we look at percent mortality for permethrin only, a main takeaway here is you can see there's a range, right? So there was variation from one population to another. The highest mortality, 96.2%, the lowest mortality, 1.5%. All right, then if we look at the numbers for permethrin plus PBO, so here we've added the synergist that helps to inhibit those oxidases. Good news here, mortality increased in all cases, and in almost all cases, mortality went above 90%. One exception here, that Winters population that I already showed you, where mortality did not move over 90%, even with the addition of PBO. Okay, so from the first goal, we found, yes, we do have resistance. In fact, it was probably a bit more than we expected. So next steps then were look to see what was happening in the mosquitoes that was leading to that resistance. First step, the enzyme testing, looking for enzymatic resistance. So to do this, the mosquitoes get ground up, and that ground up mosquito or the homogenate gets added to these plates in triplicate. The enzymes, those detoxifying enzymes that we're trying to measure, are found in that mosquito homogenate. Then you add a substrate or other reactants to the plate. This starts a chemical reaction that produces a product that has a color. So depending on how much enzyme activity there is, you get kind of a darker or a lighter color, and that can be measured using an instrument that measures the absorbance. So it measures how much color or how much product is in that plate. We tested for these groups of enzymes: oxidases, alpha esterases, beta esterases, GSTs, and acetylcholinesterase. We also did a general test for how much protein was there, and that helped us normalize for the size of the mosquito. So these box plots are how we show the data from those enzyme assays. Let me just take a minute to walk through this first one. So on the y-axis, we have the amount or the activity of the enzyme. In this case, we're looking at alpha esterase. It's divided by the amount of protein to help normalize for the mosquito size. Across the x-axis, we have different populations. BFS on the left is our colony, and then this is some of the San Joaquin County locations on this particular graph. For the box plot, in the box itself, basically that represents the amount of enzyme for 50% of the population. If you go all the way to the whiskers, that takes in most of the population, and then you'll see some dots above those whiskers. Those are outliers, so mosquitoes that are expressing a lot of these particular enzymes. One more thing to point out on this graph are those letters. Those letters denote statistical significance. So if a particular box has a different letter, then it may be statistically significant from the others. So specifically about alpha esterases, the first thing you'll notice is that our colony levels are high. not at all what we expected and not at all good news. Basically, what that means is that our susceptible colony that we had hoped to use as a reference actually had much higher levels of esterases than we expected. You can see some trends from the populations here. So Empire and Vine, for example, have higher levels of alpha esterase than the other populations shown. We see a similar thing when we look at the beta esterases, not too much difference in the wild populations, though Vine is maybe a bit higher than some. The BFS colony, though, by far the highest. Again, this is not what we expected and really leads to the unfortunate takeaway that our colony is not good, is not a good comparison colony when looking at this data. Moving on to oxidases, now we expected oxidase levels to be high, and we know that oxidases are at play because of the bottle bioassay when we added the PBO. We didn't quite see that, but again, we're struggling with how to interpret this data because of the issues with the colony. So putting that to the side, we can see a couple of populations where the mosquitoes had higher levels of oxidase, namely Vine and Empire. Similar trend to oxidase for the acetylcholinesterase. We can see that Vine population again has quite high activity for acetylcholinesterase. All right, so without having a colony to compare to, that's how we had planned to do all of our statistical analysis. But because of what we found, that type of analysis isn't going to work for us. So I'm currently working with a statistician to figure out different ways that we might analyze this data to see what information we can take from it. One thing we're doing is looking at regressions. So here are some really preliminary regressions. So I wouldn't want you to leave saying this is absolutely the truth, this is what's happening, but this is a work in progress. So if we look at these regressions, we have survivorship on the y-axis. So higher on the y-axis means more mosquitoes survived, or there's more resistance from those bottle bioassays. And then on the x-axis, we have enzyme levels, or a measure of enzyme levels. What we can see is for acetylcholinesterase and the GSTs so far, there is a significantly positive correlation. So as those enzyme levels go up, so too does survivorship. And it's near positive for oxidase. So we are working to add more populations to this analysis so that we can get kind of a more robust or a better feel for how these enzymes might specifically be affecting resistance. Moving now to the target site mutation. So this again is that DNA mutation. So for this we have to do molecular assays. It's specifically a cybergreen qPCR. And we have 3 different primers that match up to the 3 different alleles we're looking for, that leucine, phenylalanine, and serine. The underlined letter here is the DNA mutation that distinguishes those 3 alleles. And then we add those tags that you can see in green. That basically just helps us differentiate them. I'm not going to go into the details of how this assay works, but here's what the output looks like. So the takeaway for you here is that based on where that peak is, we can tell if it's phenylalanine, if it's leucine, or if it's serine. For those of you who may be interested in this assay, right now it can't differentiate a leucine serine from a serine serine, but that's a pretty rare combination, and we do have a secondary test that can help with that. I'm just going to show you a bit of the data that results from this assay. So here are some of the populations from Lake Placer and Saculo. On the left, we have that KNWR colony. This is our susceptible colony. Good news, all of the alleles in that colony are the leucine or the wild type or the susceptible allele. So the green here, leucine, is our susceptible allele. So that's what we expect. That's good news. Not so great news when we look at the rest of the population. So for each population, you can see a small green bar at the top usually, or maybe not a green bar at all. That's the susceptible allele. Then we have serine and phenylalanine shown in the kind of yellow and red color. Those are our KDR, our resistant alleles. So the main takeaway here is that in many of our populations, in most of our populations, those KDR alleles are more prevalent than the susceptible allele. So while my grad students were working on this project, I was also teaching a class, and I was looking for a project for those students to do. And I thought, what data do I have, or what mosquitoes do I have that I might be able to look to see how long KDR has been prevalent? So I dug into the freezer, I found some Culex tarsalis that I had from my days in grad school, and I was able to pair up, or closely pair up, a couple of sites. So in this slide, on the left-hand side are samples that were collected in 2006 to 2008. On the right-hand side, samples collected 2016 to 2018. Two sites are nearby sites in Yolo County and nearby sites in Sutter Yuba County. The takeaway here is that 10 years ago, 12 years ago, give or take, there were far more susceptible alleles in our Culex tarsalis populations than there are today. So to summarize, there is substantial resistance to pyrethroids in Culex tarsalis in our study counties. Mortality ranged from 1.5 to 96.2% in those bottle bioassays. And the addition of PBO brought mortality above 90% at most sites. The enzyme data is a little cloudy because we don't have a good colony to use as a reference at the moment. However, based on those regressions, there's a potential that increased oxidases, GSTs, and acetylcholinesterase might be leading to resistance. Finally, we have a high prevalence of those KDR alleles, particularly the leucine to phenylalanine mutation. There are some serine mutations as well. And the prevalence of these alleles has greatly increased in the last 10 years. With that, I say thank you and take questions. And also, if you think you might have susceptible wild tarsalis, I would absolutely appreciate you reaching out, because we're hoping to find a new susceptible colony.
Great. Thank you so much for that presentation. I do see lots of questions coming in. We're not going to get those right now. We will catch those all in the Q&A panel discussion at the end of the rest of our presentations. We're going to move on. So next we have Scott Kranz, the administrator at the Office of Mosquito Control Coordination with the New Jersey DEP. And he's going to present floodwater mosquito control in New Jersey freshwater species. So Scott, take it away.
Okay, can you hear me?
Yes, we can.
Did my PowerPoint show up yet?
Right now, we just have your Windows screen up. There we go. There it is.
Okay, great, thanks. All right, well, it's a pleasure to be here this morning. And I wanted to thank Leanne and the Veiling crew for making this event possible, this Floodwater Mosquito Control Summit. And I'll also note that floodwater mosquitoes are by far the most troublesome pest species that we deal with on an annual basis here in New Jersey. So my objectives are to review the species using a concept of life cycle types for our New Jersey mosquitoes. I'll then touch on the organization of mosquito control in New Jersey, and then move on to a couple of the state programs and some contracts that we use to help aid the mosquito control efforts here in New Jersey. The goal there is to set up for the talks, and New Jersey talks are they're gonna follow. So we proposed this concept of species life cycle really as a teaching tool for mosquito biology. And what we realized is if we group the different mosquitoes that we have in the state on these 4 criteria, where the eggs are laid, the typical larval habitat where the mosquitoes develop, the number of generations per year that the mosquito goes through, and then the life stage that overwinters, you get a fairly concise and condensed grouping of the mosquitoes here in the state. And a number of the species share these characteristics, which once you learn one of them, the life cycle type for that species, for example, it makes it a lot easier to kind of group all the other ones that share those characteristics. So in New Jersey, when you do that for our 64 different species that we have here, you wind up with 11 groupings of shared characters, and then 3 monotypic groups. In other words, a single species that fits into that life cycle group. So when we do this for New Jersey, this is pretty much what you come up with. What I'm showing you here is univoltine aiding life cycle types, multivoltine, and then as you read across, there are 5 different categories, and within that you have your Aedes stimulans life cycle type and the various species here in New Jersey that all share those characteristics, Aedes obturatus and those species, Aedes canadensis, and those 3 species. Now, when you further divide this with respect to this talk that's coming up, they all share the same or similar oviposition habitat, larval habitat, generations per year, and of course the stage that the mosquito uses to pass the winter months. So looking at this for the floodwater species more in general, what you'll notice is they're kind of restricted to that univoltine aiding life cycle type and the multivoltine aiding life cycle type. And you'll hear a little bit more about how to control these mosquitoes in the talks that follow me. When you get to our fresh floodwater mosquitoes, really there's 3 life cycle types that we deal with here in New Jersey that I'll be addressing from the state perspective. Knowing that ignores our saltwater floodwater mosquitoes, which is not a very good idea. I wouldn't recommend doing that. But we have our Aedes stimulans life cycle type, Aedes vexans, probably the most common with respect to the problems and the number of species that fall within that grouping. And then of course our Aedes canadensis life cycle type. So, and our previous speaker touched on that very nicely. Floodwater mosquitoes all have these eggs that can dry down. We call them desiccation resistant eggs. In some cases, they need to dry down, where they'll wait in a period of suspended animation. And this could be days, weeks, or even years in some cases, depending on the species we're dealing with. We actually have some species, univoltine Aedes, for example, that require a period of freezing before those eggs are ready to hatch. But nonetheless, when conditions are right, either following heavy summer rains and or tidal events, all of these mosquitoes hatch in what we call broods. So they're all developing at about the same rate. They're all larvae, all about the same size. They all pupate, very close to one another. And then they come off as blood-feeding adults within a few days. And most of our floodwater mosquitoes here in New Jersey are avid human biters. So a big nuisance, something that, the control programs would definitely need to be concerned with. And you may have noticed on scanning that list, and then from Barry's talk that started off the summit, that basically there are 2 genera these floodwater mosquitoes fall into. Now, I'm saying that with the caveat that we're ignoring Ophthalatus as a genera here in New Jersey. So they're largely restricted to your Aedes and your Seraphra species. And if you get a chance to go back and look at that list, you'll actually pull that out. It comes out very nicely. Now in New Jersey, mosquito control is mandated by state law, and it's for 3 primary reasons. Mosquitoes transmit disease, they cause nuisance, and when those populations go unchecked, they have the ability to negatively impact the economy of the state. Now these laws were passed back in 1912. And at the time, malaria was one of the one of the pathogens that was kind of driving the creation of those laws. But the more recent West Nile virus, dengue, chikungunya, Zika, serve as reminders that there is a public health nuisance perspective to mosquito control, and it certainly warrants these mandates and mosquito control at the county level. So with respect to the organization of mosquito control, each county in New Jersey has a program. Again, they're required to have a program. We also have the State Agricultural Experiment Station, in this case, Rutgers University, that plays an active role. And then we have the state in general. I'm listing here our State Mosquito Control Commission. which consists of representatives from our departments of health, DEP, agriculture, and of course the experiment station. And together, these three entities provide the balanced professional approach for mosquito control, with the counties doing the work, in large part the majority of the work, the agricultural experiment station providing research, reviewing county plans and estimates, making recommendations and providing training. And then the state responsibilities include advising the governor, providing funding, guidance, and support. Now, as touched on earlier, and you'll find this in the presentations that came before me and the ones that follow, rain and tides are the primary factor driving mosquito production here in New Jersey. And monitoring these local patterns helps determine the need for service. And you'll typically find that following these events, 8 to 10 days or so, you'll see an increase in complaints and trap counts by far. So very, very predictable once the weather events or tides have actually happened. Again, floodwater mosquitoes. With respect to the state programs, I'll start off with the first one that our state is involved with, and that has to do with surveillance. We provide funding to the university to provide weekly adult mosquito and vector surveillance reporting. They do this every single week. The data is supplied by the counties. It comes in through our Jersey Serve platform now, and it gets prepared into these weekly reports. It serves as the data, for the county programs to help make control decisions. We also provide funding to both the Department of Cape May and the Department of Health labs to provide the actual testing of mosquitoes that the counties collect. Then we have two other programs, insecticide monitoring and West Nile virus genotyping project that we're trying to get off the ground. But the important part there is we have this surveillance data and it's used to make controlled decisions out in the field. The other second state program that I'll talk about this morning is our aerial operations program. The state puts together a contract for both fixed wing and rotary wing aircraft to provide surveillance, observation, and application services upon request by the counties. Now, this program was established in 1949, primarily addresses our Atlantic coast counties. The vast salt marshes. Those counties that participate in the program would make a request to the state for services. Within 24 to 48 hours, we would make arrangements to have a ship out there to meet the need that's being requested. More recently, we've expanded this program to address fresh floodwater mosquito species. And the best example of that is our Essex and our Morris County programs. The main factor limiting the services are, annual funding, as you might expect. Now, an aspect of the program that goes along with this is our state insecticide contract. We put that together mainly to service the state operations, but we make that contract open to all of the counties. And the goal there is to provide quick, easy access to all of those insecticide formulations that you heard Elmer talking about. Goal there is you make the order and within 48 hours, the material shows up where you need it, so you can use it. A third program we have that falls into, really heavy use for freshwater mosquito controls is this equipment use program. And here we have lease agreements with the counties where the state buys these large pieces of equipment that the county uses, to address a large floodwater problem, for example. We're talking about long-reach amphibious excavators and so forth. In this case, the county maintains the equipment. When it comes into large repairs, the state provides some resources for that. Once that project is done, that equipment becomes available to another county that might be in need, so it can get reassigned. As the equipment ages, and a lot of our equipment falls into that category, currently, we can get rid of it through the auction process, to get rid of surplus equipment. But the program in general is a fantastic opportunity for the state to test new equipment. So when something new, really expensive comes on the market, state can use its resources to bring that on, assign it to a county, where it then gets tested to see how well that equipment works for mosquito control. So a very good program in that regard. And then the last program I'll talk about has to do with our efforts regarding insecticide resistance. Now, what we did is we partnered with all the counties, CDC, the Northeast Vector-Borne Center of Excellence, and the Department of Health. And what we did is we reviewed all the supplies needed to run those CDC bottle bioassays that you just heard in the previous talk. We addressed everything from egg collection to larval rearing to actually running the tests. We brought everybody together over the past 3 years for these annual workshops. And in this case, we provided all the training or hands-on efforts that might be needed. We provided some supplies for the counties to use if they didn't have them. The CDC provided the kits and the technical insecticides that we used for those CDC bottle bioassays. All got together, ran the test, and then shared that data. If counties didn't have mosquitoes available, during the workshop on that specific day, they left with treated bottles to use on their own. We did have a 2020 COVID-19 break, unfortunately, but we do have plans to continue these services. And again, the overreaching goal there is to provide these, to extend the life of these public health pesticides. And I think with that, I'd like to acknowledge all the cooperating agencies in mosquito control in New Jersey and leave a little bit of time for questions and or to move on if that's what we need to do.
All right, thanks so much, Scott. Yeah, we do still have some questions coming in, but I think we're so close on time, we're going to keep moving and we'll get to those in the panel discussion. So we'll go ahead and move on. Next, we have Mike Sinek, the superintendent of Ocean County Mosquito Control in New Jersey, and he is going to present aerial larviciding for floodwater mosquitoes of the salt marshes of Ocean County, New Jersey. So Mike, if you want to share your screen and take over, we'll go from there.
Hey, thank you. Thank you very much for the opportunity to speak this morning. Let's get here. Okay, here we go. So this is basically a 15-minute crash course of one of our largest aspects of our program. So I hope you all can follow along. Getting right into it, we're operating out of Ocean County. We're located in the southern half of New Jersey. We're second largest county land-wise, 638 square miles of land. And we have 45 miles of coastline along the Atlantic Ocean. Our salt marsh, we have approximately 28,000 acres of salt marsh in Ocean County. Now, thankfully, not all of this breeds mosquitoes. We've had a very active open marsh water management program that has knocked the acreage down. We're probably looking at really something under 2,000 acres that breed mosquitoes right now. So here's the salt marsh. It may look beautiful, but as we all know, those that visit salt marsh in the summertime, if you're this deep into it, you're usually getting bitten by something. So I'm going to talk about salt marsh zones during this presentation. What you need to know, get the laser out here. We have low marsh and we have high marsh, and it is separated by the mean high water line. Below the mean high water line is usually too wet to breed mosquitoes. All your action is going to be generally above the mean high water line. And what's really, really nice is we have a marsh grass that grows right at this union called Spartina patens. This is kind of like your cheat sheet. You can, if you can find the Spartina patens, you know where to start looking for mosquito larvae. This is what it looks like. It's commonly known in our area as salt hay. And what's really, really nice, if you're doing an aerial survey, you look down, the Spartina patens actually shows up in these really nice light green patches. So again, it's kind of like your cheat sheet. You know there's a change in elevation here. There's a change in the way that the either the tide comes up, the flood comes up. You know this is a good spot for you to start looking for your mosquito larvae. Three main factors that influence salt marsh flooding are tide, rain, and wind. I'm going to talk about them a little bit individually here. Tide is usually our biggie. You can expect the marsh to flood. You can expect the marsh to flood about twice a month around the peaks of the full and new moon phases. These are referred to as spring tides. have nothing to do with the time of year. They refer to the extreme rise and fall of water during the full or new moon. Our second factor is rainfall. Now, some of you might be saying, okay, rainfall, that's freshwater, right? Well, there's enough salt out on the marsh, on a dry marsh. Salt is being excreted by plants. It is also dry. It's dry on the marsh mud as well. So an inch or more of rain hitting the marsh and washing into any kind of depression over there is going to trigger a brood of mosquitoes. Summer thunderstorms, which are probably the worst, they can unleash heavy amounts of rain in a very short period of time, and they're a true mosquito maker. Rainfall broods are also really problematic because the actual amount of rain can vary over large areas. So what we have actually 19 rain gauges set up and down our coastline so we can monitor that. But again, rainfall is a huge problem for us because again, we don't even know when it's coming. So a third factor I'm going to talk about is the winds. This might be a surprise to some people, but high winds blowing over the open ocean or an open bay for an extended period of time can actually push enough water to flood the marsh. These winds are generally out of the east. And again, if you see the picture over to your left, that there's a situation where the marsh is being flooded just simply by a couple days of a northeast wind. So our big three species of our salt marsh is Aedes contator, Aedes sollicitans, and Aedes tanurhynchus. Here's a classic ball of larvae, not something that you want to see when you walk out to inspect. Aedes contator, their larvae are found on the high marsh, particularly at the tree line. This mosquito can be an aggressive biter, but is often overshadowed by other species. And the reason why is because of their seasonal abundance. You generally have a lot of them in May and June. So it's a late spring mosquito. Don't see much of them at all in the summertime. And by fall, you can get maybe an isolated areas of some of them popping out. But again, generally in May and June is when you see this mosquito. Aedes sollicitans is our biggie. Their larvae are found on the open high marsh to the tree line. They're a major nuisance species in the summer, and they can be tied to eastern equine encephalitis transmissions in horses and humans. Here's the seasonal abundance of them. Again, mostly a summer mosquito. If you have a very warm fall, they can actually linger late into the fall. And lastly, we have Aedes tanurhynchus. Their larvae are found on the open high marsh to the tree line as well, found right in with Aedes sollicitans, but in smaller numbers. Now, they were once only found in the extreme southern end of Ocean County. Now they are throughout all the way up to the top. So they're becoming more established each and every year. So we're going to move on to salt marsh control. Before you need to control, you need to inspect. So we have roughly 80 inspection sites up and down our coastline. A Bell Jet Ranger helicopter is used to get our inspectors out there, and we're just hopping along spot to spot with our dippers, checking any kind of depressions in a designated area. Our inspections are made at least twice a week from mid-April to early October to give you an idea of how long our season is. When controlling salt marsh mosquitoes, I kind of broke this into three categories. We have liquid larvicide, single brood. This is used on the open salt marsh where low growing vegetation is present. We have granular larvicide, single brood, which is used at the marsh edge or at the tree line where tall growing vegetation is present. And we have extended release larvicide, which is used in special situations that are unsuitable for single brood. I'll get to that shortly. So for our liquid larvicide application, our larvicide we're currently using is Vectobac 12AS. The active ingredient is BTI. The rate we are spraying at is 32 ounces an acre. That we get about a 90-foot effective swath, and our application height is 10 feet above the ground. We are targeting here Aedes sollicitans and Aedes tanurhynchus larvae at the first and second instar level. it's really effective if you can get these mosquitoes at those early instars. BTI seems to work really, really well. Here's a quick look at our liquid spray system. Again, we're using a Bell 206B Jet Ranger with a simplex liquid spray system, which is located on the belly of the craft. This also consists of long spray booms with nozzles, and currently we're using an RA4 raindrop drift reduction nozzle. So here's where you would apply a liquid larvicide. It's the open salt marsh where the plant life or the marsh grasses are low growing. Really small depressions are what we call potholes within the salt marsh grasses. And you can see the reason why we call them potholes is they actually look like something you would see on the roadway, just small depressions within those grasses. Moving along to granular larvicide application. The larvicide we're using for this is Vectoprime. The active ingredient is BTI and methoprene. And we're spraying this at a rate of 4 pounds an acre. We're getting a swath of 75 feet out of this. And our application height is about 45 feet. We call that tree top level. Here we're targeting all 3 species, Cantata sollicitans and Panurhynchus. Our granular spray system is pretty unique. It's an FAA approved self-fabricated system, which what we call a hopper, which stores the pesticides. It sits on the sides of the helicopter, actually sits basically where the passengers would sit in the back of the helicopter. And it also consists of a short pair of booms to deliver the swath. So with granular larviciding, again, you're targeting these tall vegetated areas. You want those granules to penetrate any kind of canopy that's there, let it trickle down, get to where it needs to go. Really looking for these low areas within the higher vegetation. That's where you're going to find quite a bit of mosquito larvae. So extended release. This is some of the interesting situations that we come up with. Now, obviously, this has got to be an area that is known to produce mosquitoes. That's number one. Number two, sometimes you have areas that are hard to inspect by foot and hard or dangerous to inspect by helicopter. Again, this is a typical area over on our left here where really difficult to get in here by foot, dangerous to land in here. We know it breeds mosquitoes, and we figured, okay, we need to still treat this, but we have to put this almost on a schedule. There also could be areas where entry or application period is restricted. It could be an environmental reason. It could be something to do with human activities as well. So what we're using in this situation is Metalarv. The active ingredient here is methoprene. We're spraying this at a rate of 4 pounds an acre with a swath of 75 feet. The application height, again, is 45 feet or treetop level. And here we're targeting sollicitans and Aedes Panurhynchus. Now, when you're monitoring your treatments, as far as single brood goes, with Vectobac 12AS, you can back check in 24 hours after the treatment to determine the larval mortality. With Vectoprime FG, you can back check in 24 hours, but you have to remember there's methoprene also in, and it's dual active. So if you find any living larvae, you should really be observing them into the pupal stage. Now with Metalarv, if possible, and I know some of us, if you're using this, you can't really access the area, but if you can get in there, the best thing to do would be to collect the pupae and monitor them in an emergence jar. If the area cannot be monitored, you might have to resort to something like landing rates, or you can set traps. In one area, we actually set a CDC trap, which is baited by light and CO2. And you can keep this out there during the 42-day period when Metalarv is effective. And it'll give you a pretty good idea of what's coming out there, if hopefully nothing at all. But again, this is something that we have to resort to if we can't actually go in there and make a collection. So one of the final things I want to say is about the salt marsh is what you consider a dynamic environment. When you're controlling mosquitoes in a dynamic environment, you have to realize that the salt marsh is always subject to changes brought on by extreme weather events, sea level rise. This affects the flooding patterns and also the areas where mosquitoes can breed. You see on the picture down on the left there, massive tree die-off from saltwater intrusion. There's something going on here. There's a change here. So we need to keep our eye on this area. On the right, we have a picture of Phragmites die-off. Again, this area is staying wetter than it should be. It's killing off those Phragmites. So it's letting us know that there is a change in flooding pattern here, and we need to keep an eye on this area. So with that, I thank you all for listening. This is our aerial larviciding staff. There's only 4 of us. We get a lot of work for these guys. They get the job done, and they do get some help along the way. But great job that these guys are doing, and I really appreciate it. I appreciate all listening to me. Thank you.
Thanks, Mike. Thanks so much for that presentation. I'm going to give you one quick question here that we have for you.
Sure.
I've always heard that heavy rain will actually drown mosquito larvae. Is this true?
Now you have to realize that mosquitoes are not mosquito larvae are not the best swimmers in the world, so they can maybe flush them out. I wouldn't say drown. Usually they are going to, the water's going to come up and they will go to some area, push back farther. As long as the fishies don't go in and get them, they might be able to find a corner and they'll be okay. No, not drowning. I mean, they do have to breathe.
Yeah.
There, but
Just move them to another spot. So.
yes.
All right, great. Thanks for that, Mike. We're going to go ahead and move on to our next presenter. There isn't a couple more questions, but we'll come back to you in the panel. So, all right, up next is Josh Kogut, the Director of Aerial Operations, and Mickey Koss, the Director of Operations for Florida Mosquito Control District. And they are going to jointly present Florida Keys Diverse Operations and Response Methods. So looks like Mickey, maybe you're up first if you want to get going. Or Josh.
I'm working on it.
All right, excellent.
All right, my name is Mickey Koss. I'm with the Florida Keys Mosquito Control District. I'm the Director of Operations. I'm here to talk about the diverse operations throughout the Keys. The Florida Keys consists of over 800 islands stretching over 180 miles. Each of these islands are affected by tide in some way or another. We have 42 bridges throughout the Keys, and 2/3 of Monroe County, the Florida Keys, are made up of wetlands. Florida Keys Mosquito Control has partnered with many organizations throughout Monroe County. You can see here with the checkered red, those are all no spray zones throughout the Keys, which makes our organization more larval control than anything else. We partner with the U.S. Fish and Wildlife, we also deal with the State Park Services, Navy, and of course, private properties. The U.S. Fish and Wildlife consists of both the Great White Heron Wildlife Refuge and also the National Key Deer Refuge. They're located in the Lower Keys. You can see here in the red, they're owned areas of the U.S. Fish and Wildlife. All of the offshore islands that are not connected by the bridge are also areas that we are allowed to go out and inspect and treat with BTI only. There's many endangered species throughout these areas, including the key deer, the white rice rat, the Lower Keys marsh rabbit, and multiple species of butterflies. Because of the endangered species, we have worked out an agreement with the Fish and Wildlife and their protected lands. You can see in the map on your left, the blue areas are non-citizen parcels, which are managed by the U.S. Fish and Wildlife. And of course, the small portions of yellow, which are privately owned property. To be able to treat these properties effectively it would not work at all. So we did on the map on the right, work out agreements of being able to treat particular neighborhoods with fog trucks. Basically in green are the areas that we can treat with fog trucks. The red lines are the areas that we were able to treat with fog trucks, but are no longer parts of our routes. Next, we work with the state, Florida State Fish and Wildlife. Highlighted in yellow are parcels that are managed by them. They're on many islands throughout the Keys. They're also owned by nature conservancies throughout the Keys, but are managed by Florida Fish and Wildlife. Many islands throughout the Keys have these parcels, and each island has a different agreement and the threshold of when can be treated for adult mosquitoes. We also work with the state and national parks throughout the Keys. We have 15 areas. Each one has a varied agreement and lists of products that can be used on that property. And of course, next we have the Navy. We have Boca Chica Naval Air Station and also the Key West Navy bases. Here we're showing the traps and landing rate counts throughout those installations. In Key West, you'll see 4 separate areas in and around Key West, which hosts multiple defense agencies. Key West Navy housing is also a separate entity in those areas in Key West, which is also a different contract with different reporting and product contents. And next, of course, we have the privately owned properties, which of course is per label. These are all the challenges made out through the Florida Keys. To answer this, we have 32 field inspectors throughout the entire Keys. Each inspector has their own designated area and treat everything within that area, whether it be from tides, the rain barrels on people's properties, solution holes, drains, anything within their area. Their biggest job is public relations and teaching the residents how to take care of their yard. And they also make personal relationships to gain access. Sometimes domestic inspections could be known as garbage collectors because they're picking up tires and turning over water holding containers in each person's yard. And we have 4 offshore inspectors. They're treating both private islands off the beaten path and offshore islands that are owned by, managed by U.S. Fish and Wildlife. The offshore inspectors are setting traps, collecting. They're also IDing all the traps in the lower Keys and the larval collections brought in by all the inspectors throughout the lower Keys. And of course, we have the 4 top research entomologists and biologists. They are in Key Largo, Marathon, and Key West. And we do have our other components to wrap up our great organization. All of the inspectors and biologists, office staff, we all use Field Seeker, both the desktop and the phone application. In the office, we create reports of all the data that's entered. We monitor and enter service requests. We can check the ring gauges and landing rate counts live. Inspectors will enter all their counts and ring gauges and all their treatments, trail maintenance, truck maintenance, enter their public relations and anything that they do out in the field. They also can put proposed treatment areas for future larvicide or barrier treatments. They can select any known polygon or a portion of it. They can create an entirely new polygon or polygons to treat by selected due dates. Proposed aerial treatments are set in a staging area and then are responded to by the aviation department. which leads me to Josh Kogut, the Director of Aerial Operations, and he will discuss the diverse aerial operations from that task.
Thank you very much, Mickey. Let me just get the screen up here. Okay, thank you very much for having me. Is everybody okay and hear me?
Yep, everything's good.
Great. Today I'd like to talk to you about some of the diverse operations here in the Florida Keys. As Mickey stated, we have some of the most unique geographical locations to conduct mosquito control operations, and obviously that rolls over to the aviation department in those challenges. We'll be talking about some of the typical days here in the Florida Keys in terms of our treatment areas, what tidal and floodwater activity does to those typical days for us, and some of the tools that we utilize to combat those problems. As Mickey stated, we have a very large, lengthy area in Monroe County here in Florida. Throughout this area, we operate 5 different landing zones and loading zones for our aerial department. And we are located in Marathon, where our central location is. All of our aviation assets are located here in Marathon. One of the biggest challenges we have is really getting to the treatment areas throughout the day as proposed treatment areas come in, just given the geographic distances between several of the areas. Starting off here in Marathon is our home location, as I stated. We have 6 helicopters and 2 fixed-wing aircraft that we operate out of Marathon. Going back to those distances, we operate from the central going to the north. Our first stop, loading location, if you will, is 33 miles away. So in order to even get to some of the areas, we're traveling for 20 to 30 minutes by air just to get to where we're treating. Here we operate out of Tavernero Park. It's a small airstrip up to the north. Going further up to Key Largo is our highest location to the north. And that's going to take another 15 miles. So if we are to go from Marathon to Key Largo to treat just for a single day, we're looking at least a 30 to 45-minute trip there and then another back. So it can get pretty interesting. Several locations we operate out of are dead-end roads, small areas throughout the Keys for loading zones. We are geographically limited in the amount of real estate left over for landing, just given the population down here is increasingly getting larger and larger. So it's difficult to find loading zones. Going to the south, to our Big Pine location, our Lower Keys location, we have another 22-mile trip to get down to there. Operating again off small dead-end roads for loading zones. Continuing on to our next location is Summerland Airport we operate out of. That's an additional 23 miles down. And then finally down to Key West will take us another 20 miles. So again, going from our central location down to our southernmost point can take a good 35 to 45-minute flight. And this is Key West Airport, which we often operate out of. Looking at an average summer day for us, we typically have about 800 acres of granular larviciding. That's kind of a typical treatment day for us. We would usually utilize 2 aircraft for those kind of days. This is basically rain and water only, rain events only. This is not any kind of tidal activity. When we do get tides in conjunction with our standard rainfall in the summer, days like this can easily go up to 2,100 acres, requiring me to have 3 aircraft operating throughout the Keys. This is just the Lower Keys, and this is a typical day. You can see the flight paths of all the aircraft and the areas that we've treated through our tracking systems. One of the ways that we were able to do this is through technological advancements, better technology and better equipment. So here is a picture of one of our newer H-125 Airbus helicopters. We utilize an AgNav spray system you can see in the lower right-hand section of the picture, and some of the most advanced GPS navigation systems and tracking systems we have. One of the ways that we are able to do this so efficiently is active flight following and wireless remote data transfer. So Mickey, my associate, had touched on the fact that we utilize Field Seeker software. So they're able to take Field Seeker, enter PTAs for us to treat, and we are now able to send them directly to our helicopters without having to actually plug anything in, take anything with us, USBs, et cetera. So we can actually do it through the air, which makes sending PTAs to my helicopter pilots very fast. Again, this is kind of a picture of a standard day showing some of the tracks for treatment areas. In the center there, you'll see where we have a central loading area. So going back and forth, you can see the amount of trips it takes to get from the loading area to the areas we treated them back to that loading area again. This decreases the efficiency of the spray operation. So the less times we can travel back and forth between the treatment area and the loading zone, obviously we can treat more acres fast. How this works is you'll have a field inspector in the field, as Mickey said, testing and then entering that data into the Field Seeker software. It can then be transmitted via the cell servers down here to our flight follower or dispatch area, depending upon who that person is for the day. We can then take that proposed treatment area request, transfer it over to our software utilized in the aircraft, our AgNav software, and that can be sent back again to the helicopter, to the aircraft or the airplane. And from there, the pilot will receive a message on his screen saying he actually has mail. Actually gets a blinking sign there up to the right-hand corner saying you've got mail. And then from there, the new spray area is downloaded into the aircraft and the pilot can begin entering it. We utilize a 4G system and enhancement system for the cell service down here, given the kind of remote geographic locations. So we have what's called a hotspot in all the aircraft, which downloads or assists us in downloading cell service to the aircraft, which is then sent via Wi-Fi to the aircraft system itself. Sure thing, I'll wrap things up here. The aircraft, basically, we treat in 3 areas for this day. If somebody in the field were to add a real-time treatment area, we could actually take that immediately and send it to the aircraft, and therefore the aircraft would not have to deviate to go back and get another load. They could just keep spraying as they receive new things. Last but not least, I'll wrap things up. Previous days of loading used to look like this. Several people in the field helping to assist the helicopter in loading. It could take upwards of 20 to 30 bags to load our aircraft up. Our new system that we built in-house is a 6,000-pound granular loading system, and it sprays or loads the aircraft directly through air, and we can fuel at the same time. So we're able to get in and out of a loading zone in about 8 minutes for a full load and with fuel. So this greatly increases the amount that we can do per day. Thank you for your time.
Awesome. Thank you very much, both you and Mickey, for your presentation. There are some questions that we'll wait till the panel get to them just because we're so close on time. Just to let you all know, you probably have seen it in the chat. The pop-up quiz number 2 has shown up there. So go ahead and take your quiz number 2. Again, those states are California, Utah, Colorado, and Oregon. So make sure that you go ahead and take your quizzes. We are halfway through now, so we are going to take a quick break, about a 10-minute break. We're going to go ahead and do what you got to do, and we will come back at 10 after 11. So we will see you all in 10 minutes. All right, we have about 1 more minute to the end of break. So come back here in 1 minute and we'll get started. All right, everybody, we should be coming back from break now. It's 10 after 11 Eastern time, so we're going to go ahead and continue and get started again. All right, so next on the agenda is Mark Clifton, Executive Director of North Shore Mosquito Abatement District in Illinois. And Mark is going to present wide area larviciding with a buffalo turbine and Vectolex WDG. So Mark, you want to share your screen and take it away?
Did that work?
That did work.
All right, terrific. So thank you for having me. My name is Mark Clifton. I'm the Executive Director of the North Shore Mosquito Abatement District.
One thing, you're not in presentation mode yet. We can see all your others.
Oh, you can, okay.
Oh, we lost
How
you.
about now?
Now we're perfect.
Okay. So I am the Executive Director of the North Shore Mosquito Abatement District, and today I'm going to talk about wide area larviciding with a buffalo turbine and Vectolex WDG. Typically, you see this kind of larviciding with Vectobac WDG, especially for container mosquitoes. And we thought it would be interesting to try this for floodwater habitat, especially here in our kind of unique district. So in case you're not familiar with where the North Shore Mosquito Abatement District is, we're located in northeastern Cook County, and that's in northeastern Illinois. And this is actually Chicago. We're a suburb community of Chicago. We range from really dense urban all the way to suburban habitat. Through the center of the district runs the the north branch of the Chicago River. And on either side of the river are extensive floodplain and green areas that have all been preserved as parks. And you can see that in the picture in the top left. All that green space can all flood. It's all along a river. So if you know anything about Chicago, it's notorious for rainwater and floodwater issues, has been for a long time. And in the early spring, these areas will fill up. You'll see in the center pictures, we treat a lot of this by manually with a backpack. Our staff walk through this deep floodwater treating with either BTI or Bacillus Sphericus. And the picture at the top shows how dense a lot of this habitat can be. It can be really impenetrable. Typically, we would treat this with a vortex. That's the picture on the right. A lot of these green spaces have running trails through them. And so we would take a truck and kind of shoot granular material into the woods, hoping to get enough into a lot of this floodwater. And as you can imagine, it works better in some places than in others. The vortex is a great tool, but some of these floodwater areas are quite large. So this is a real problem for us because we only have a 4 or 5 days maybe between a heavy rain event and when mosquitoes will emerge. On the right is a band of 4th instars and pupae just ready to ruin a barbecue somewhere. And this is extremely common in all our flooded areas to have just enormous broods of vexins. And so the key for us is how do we get through all of our floodwater in a really short timetable after a rain event with really, limited staff. We have 12 seasonal staff members generally and 7 full-time members. So we took some cues from the Zika response in Miami and some of the other ways that Vectobac WDG are used in Florida. with the buffalo turbine and thought, well, maybe we could do this with Vectolex and Sphericus. And so we set out to kind of recreate this method for our floodwater areas. So to do this, the first thing you need is a buffalo turbine. We were fortunate to get one from Macon Mosquito Abatement District in Southern Illinois. They had no use for it anymore, and they basically gave it to us. You need a rotary atomizer. These are typically used in aerial application of adulticides or larvicides. And then Micronair also sells a adapter, which is the picture on the top right. And so it's pretty easy to retrofit a Micronair onto a buffalo turbine. Micronair has this all set up and ready to go. And then of course on the bottom right is Vectolex WDG, and WDG means water dispersible granules. So this is kind of like a pancake mix that you mix with water. And then is basically a liquid larvicide. So there's a few important things to use a turbine in this way. You have to set up the correct flow rate. You have to have the correct blade size for this Micronair. Basically, the rotary atomizer sits in the airflow and spins based on the blade size, and the blade angle will control how fast the Micronair spins, which controls the droplets that come out of the turbine. And so it's important that everything is set up just right. The right flow rate, the right blade size, the right blade angle, the right concentration, the right volume. It sounds complicated, but Leanne makes it so easy. It's really not hard at all. So Micronair has a manual which shows how to kind of organize this rotary atomizer so that it'll give you the correct droplet distribution. We know we wanted droplets at about 150 microns, and the figure on the right shows that we should want a rotational speed of about 6,000 RPM. And then the figure on the center there, to get 6,000 RPM at about 140 miles per hour, which is the wind speed coming out of the turbine, you want about a 55-degree blade angle, and that gets you right where you need to be to make this work. Then you have to calibrate, mix and calibrate the material. We calibrated this for about 3.2 gallons per minute at 50 PSI. And at 10 miles an hour, this will apply exactly, or just about, a half a pound of Vectolex per acre. So that's our final application rate when everything is calibrated and set up. This is a beautiful spring day in Chicago. It was a gorgeous day. And we set up a bunch of chrome coat cards perpendicular to our buffalo turbine and mixed in red food coloring. This is pretty standard with buffalo turbine or other kinds of calibrations. Whoops. And we basically were able to count the number of droplets and the size of the droplets to determine how far this material went and what its droplet distribution looked like. And so after we did this, we found that we got pretty decent coverage out to about 300 feet, probably got even further than 300 feet. We had a volume median diameter of about 150 microns. And then our droplet density, of course, was highest between like 100 and 200 feet, but stayed pretty good all the way out to 300 feet. So it's important that the droplets are the right size, because if they're too big, they won't go very far and all your material will end up in the first 50 or 100 feet. If the droplets are too small, they'll go too far and you'll have too thin distribution in the area you're trying to treat. So trying to find a happy medium is important, where you have enough droplets that land closely and enough droplets that land 300 or 400 feet out to get control throughout the entire area. The next step was to test this operationally to see how this worked. So we picked an area along a jogging path where we had a treatment site and a control site. And both these sites are about 1 acre in size, and they're notorious for being really productive for mosquitoes. and they just harass people as they're running through or biking through, whatever. The goal was to apply about 1/2 pound an acre. This is a picture of that, one of those floodwater sites. Believe it or not, in that brush is about, you know, a foot of water. It's just incredibly dense and almost impossible to get to with a backpack. We used GIS to pick 10 random points per week. And then we went out and sampled each of those random points. The only way we figured to really assess statistically the effect that our treatment had on larval mosquito populations was to do a truly randomized design. So there is software freely available that'll give you random points within a polygon. And that's what we used. It feels a little stupid to go where you know there's not mosquitoes and sample, but that's the only way to do it. If you're always going to where the larval mosquitoes are, then you're really biasing your sample. So it was important to have randomized points. So we would dip a single dip and then count how many larvae were in the dip. And then we sampled these for 7 weeks, 10 dips per site. And this is what we found after one treatment. On day 0 or week 0, which would be the time of the treatment, there was really no statistical difference whatsoever between our treatment and our control site. And then in the week following, there was already a statistical difference. Our treatment site had basically 0 mosquitoes in week 1, 0 in week 2, and then maybe just 1 or 2 in week 3. And this lasted for a total of 6 weeks until we ended the experiment. So we started early with this, and we didn't see mosquitoes return to the treatment site really for the whole season. I think they just were that pretty much done for a while. And every week here was statistically significant except for the very first week. More district-wide results. In 2018, before we had the turbine, we were able to do maybe 200 of these acres manually by backpack and by vortex. With the turbine, we were able to increase that dramatically to maybe 11 or 1,200 acres treated, and which was like basically a 5-fold increase in the amount of acres we're able to treat. The figure on the right shows the average weekly precipitation on the x-axis, and then the average Aedes vexans mosquitoes per night on the y-axis. And you can see roughly that there's a positive correlation that when you have more precipitation, you're going to have more mosquitoes, of course. The red dots are 2019. And that line is essentially completely horizontal, which basically says that despite a lot of precipitation in 2019, I think we had the wettest May ever in history, we just did not see big broods of mosquitoes. Once we employed this method, we were able to quickly treat the areas we needed treated. This is the first year that there's really not a lack of correlation, that there's really a lack of correlation. Every other year that we've looked at, and we have data going back quite a ways, there's almost always a positive correlation between precipitation and mosquitoes. So this is some positive evidence that if you can go out and find where the mosquitoes are and treat them timely, it will make a difference over, at least over our 70 square miles. So along the way, we came up with some other methods for mixing Vectolex. Mixing is lots of different ways to do this, but we found a stainless steel dewatering pump, the biggest one you can get that's still electric. I think it was like a 3/4 horsepower or something. And we actually just drop this into our tank, and then we'll put a funnel on top and pour the Vectolex into the funnel and let it kind of just pour into the tank and mix on its own with this dewatering pump. The advantages for us are we don't have to empty any hoses with excess material. We don't have to rinse any hoses. All we have to do is lift the dewatering pump out and put it in a bucket. We have our district is kind of in an urban area, and dealing with rinsate is not really an easy thing for us to do, and we don't have a lot of room. So this was a really great compact solution for effectively mixing this material. It worked quite well. So I would highly recommend exploring something like this if you're interested in using any of these WDGs. And then finally, On the left is our district and our Buffalo turbine, and the right was the actual treatment that we did. You can barely see it on a bright sunny day. We did this first thing in the morning, but it was very effective. So thank you for listening to my presentation, and I'll be a part of the panel later if you have questions you want to ask.
Awesome. Thank you so much,
Mhm.
Mark. We definitely do have a couple questions for you, but I am going to hit you with one real quick just because we do have
Sure.
a minute. What kind of wind direction and speed did you experience? I'm assuming during those types of applications. And was there any time that you applied without the ideal wind conditions?
You know, I think we had, during this experiment, we had just a few miles an hour. It would have been better to have a little more wind, but it seemed to be effective. We've been applying first thing in the morning. Typically this is done at night, but here, especially in the spring, we've had good results applying in the morning. We haven't really had the need to go at night when the atmosphere is different. So I think because we're further north and because the spring is cool and the ground can be cool, we can get away with earlier applications in the day. Wind speed, and you know, if it gets too windy, I'm not sure that it really does much. It just moves sideways. You may get some control beside the truck, but a lot of those smaller drops just move sideways. So if it's above, I don't know, 8 or 10 miles an hour, we won't bother.
Great. Thank you so much for that. All right, we'll save the rest of the questions for the panel and we'll continue to move on. So our next presentation is by Peter Brake, the Director of Technical Development for Collier Mosquito Control District in Florida. And Peter is going to discuss Collier Mosquito Control District's use of UAS platforms in areas of salt marsh mosquitoes. Peter, whenever you're ready.
Well, good morning, everybody. As Leanne said, my name is Peter Brake. I am the Director of Technical Development at Collier Mosquito Control District in Southwest Florida. I saw a potential use for unmanned aerial systems, also known as drones, in mosquito control for application of material and for real-time habitat inspections in early 2016, but we started mapping around mid-2018 due to some organizational changes within our district. The district was the first organization in the state of Florida to get authorization to dispense material from the drones shortly thereafter. So, is my slide progressing?
Nope, it's not. We're still on your first slide. Oh, there we go. It caught up.
Collier County is located in the Southwest Florida, surrounded by 5 counties in the Gulf of Mexico. The county is over 2,300 square miles, and just over 1,500 square miles of that is protected land from either state or federal lands. And we want to treat the area as best we can with the growing population that border the district. Collier County is over the double area of Rhode Island. Currently, there are around 380,000 full-time residents in Collier County, and will increase an additional 80,000 people in the winter. The district is founded in 1950 with 6 square miles. We are now over 400 square miles and operating with a $27 million budget. Tourism is a big part of our industry in Southwest Florida, and over 2 million visitors in 2018 spent over $1.5 billion. So mosquito control is essential in this area. So how do we do this? Well, our main platform for treatments consists of 3 Skyvans, 1 Bell 7, and 3 MD500 helicopters. The Skyvans are strictly for adult siting, and with a large volume tank holding enough material for around 37,000 acres at less than half an ounce per acre. 2 helicopters for our regular larvicide applications in areas around 50 to 750 acres, and are useful for smaller areas of salt marsh and mangrove areas for the adult siting role. We have 8 full-time pilots and 7 field technicians. But our current fleet for drones, we have a very diverse fleet of drones, with each one completing a very unique task. We have 1 Mitsu 600 or PD-13. That's an application drone that can carry 13 pounds of material the Phantom 4 NDVI has a camera filter, and that red filter is used for mapping of areas that we need to get a true NDVI. I'll explain it a little bit more later on. One splash drone is a waterproof drone that has a camera underneath for inspection under the water, fish, larvae. We can see that through the camera underneath. One quantum systems Trinity. It's used for mapping only, as it's got a 20 megapixel camera that points down. This is only fixed wing drone that we have, but it still can take off vertically like the other drones, so it's good for tight areas. And of course, we have 3 Mavic Pro Platinums. We use those for inspection and mapping. The total cost for the program is around $68,000. It might seem like a lot, but for a large segment of IPM, we can treat material in needed areas with all but one drone, and that would be the Trinity, as that's, again, for mapping only. Our drone program is designed for areas between 5 and 50 acres. Manned helicopters can treat areas over 50 acres easily, and below 5 acres, we use our ground-based platforms. The Mavic Pro Platinum is the main workhorse of the district, doing most of the mapping, habitat inspection, and leading to an increase in efficiency in the field. These are operated by using a controller and a tablet interface to see what the drone sees in real time. We are working with putting information from the drones in FieldSeeker, and the operations team can see the pictures of the wetland areas once uploaded. These can make our technicians' time more efficient, as they have the capability to see if the area is wet before trekking through the woods into the habitat, along with other snakes, spiders, and other things that wouldn't be good for them. These can be set up in a couple of minutes. We have used the Mavics at night for information to put into our guidance system on our manned aircraft when conducting testing or treatment in the areas that we don't have a wind tower at 300 feet. The drone automatically calculates the pitch and roll inputs needed to maintain a constant position, and that can be read in other software and then sent to the pilots. The Mavics can also provide a different sort of NDVI data called VARI, or Visible Atmospherically Resistant Index. And while it's not a true NDVI, the differences are minimal if you're going to set up a drone program. I can send the information later if desired. The content information will be at the end of the presentation. So the Mavic Pro is essential in increasing mapping capabilities by getting real-time information in these areas. These drones offer higher resolution than Google Earth, as it's collecting its images from 300 feet as opposed to the 22 miles from Google Earth satellites. There are some limitations if you're doing large areas, with heat being the biggest factor in the middle of the day with the tablets overheating, which is why we have the next drone. The Quantum Systems Trinity is mainly used for larger areas, over 100 acres usually. The flight software is installed on a laptop or PC and uploaded to the aircraft, and because it operates under a different controller, it can fly longer, making the mapping process more efficient without heat being a factor for the aircraft or controller. Like I said, it takes off vertically, and once it's reached its assigned altitude, the propellers on the wings transit forward and shut down, and the one on the tail there actually propels the whole aircraft for the whole 90 minutes. So we get a lot of flight time, but with a 7.5 foot wingspan, again, it can get into, it's designed for large areas, so we want to minimize the clearances with the trees. This area was flown with the Trinity. It allows us to get large areas mapped out, not only more efficiently, but quicker than Mavic Pro drones. The picture in the blue border is the area that is flown. The yellow is as it is flying, and the red is the final product with a 12-minute flight time. So we get a lot more accurate pictures of areas. In here, you can see the aquatic weeds and the canals. Over time, it's going to be good for mapping out new areas and get consistent mapping. The Phantom 4 has the NDVI camera. Again, NDVI is Normalized Differential Vegetation Index, and it measures the level of chlorophyll in the trees in areas of dead or dying habitat. Mangrove eggs are usually found where the red and green areas meet. The red or indicates die or dead, dried, or flooded grounds without vegetation. The green area indicates the green, healthy plants, and it's good to show that transition as we can do our process of eggshelling in those areas to find out where most of the Ecology will lay their eggs. Mavericks help us to get in these areas that are dangerous by helicopter or inaccessible by foot. Originally, our manned aircraft rely on Google Earth images. Soon to be Field Seeker, we produce the file format that we need. The Google Earth has information that was 12 to 18 months out of date. With this area, one of our common treatment areas in our pre-treat schedule, and we usually treat this area 3 times per year. One flight from the drone and other research work, such as the eggshelling, we verified that this block can be reduced by 87 acres. And at our current rate of £8 per acre, it saved almost $11,000 in one mission and over $32,000 for the year in this one area. We have somewhere between 100 to 150 blocks in our district currently. So the process for stitching all this together is drone deploy. This is a program that can give you the data when the tablet's connected to the controller. It plans the flight, tells the drone to take off, fly the intended route, taking many pictures and lands. Once you have all the pictures, it's uploaded to the website. In a couple of hours, you have the final product, which is the map. And with that map, we can determine whether we want the 2D map or a 3D model, which is good for elevation, and of course, the vary aspect, or the NDVI aspect as well. And once the map is generated, I can export that as a KML or KMZ to turn into our AgNav system or our Field Seeker system to convert that into a map for our larvicide blocks. With our Matrice 600 PV13, these are mainly completed by this base platform. The drone can apply liquid larvicide with about 13 pounds of material, and it gives about 5 minutes of spray time with our current flow rates. In the adult siding roll, we can treat about 200 gallons, 200 acres per tank in the adult siding roll. And with the same aircraft, I can transfer the granular application kits in about 5 minutes. This also dispenses about 13 pounds of material, which is the max capacity of the Matrice 600. We have an application rate of 8 pounds per acre, so we're often reloading after 1.5 acres. This drone is best suited for liquid and granular applications in urban environments where the aircraft will be present safety risk or noise complaints by, but we can also use the drone for adult siding roll, where it would be too small to complete by manned aircraft, and it is then uploaded into Field Seeker to see the application for our records. We're looking at purchasing a bigger drone here, and it should be arriving in the next couple of days, weeks, and it will effectively double our capacity. So instead of the 13 pounds, we'll get nearly 24 pounds worth of material. So drones offer a safety aspect as well. In this slide, the area on the left shows an helicopter application, and the lines in yellow are the aircraft flight lines. And as you can see in the left side picture, the pilot has to fly between the buildings as they do not have time to climb over the 300-foot residential towers. With this, we get a high number of both noise and safety complaints from out-of-town residents not knowing what we do. With the drone applications, we stay over the intended area of treatment and can reach out to the public and communicate with members of what we are doing as they drive by. The downside is, again, it's such a small airframe. We don't have the payload, but we have the ability to only go over the habitat and a minimal noise footprint in the areas. Drones complete many roles to assist in mosquito control programs in all departments, and it's a very effective role and tool in the IPM or IMM, and in a floatable way, gathering vast amount of data. We have many published papers out, both in JMCA and in Wingbeats. Look out for those. And with that, are there any questions?
All right, thank you so much, Peter. I'm just checking the Q&A. I don't think I see any questions directed right at you. So we'll take advantage and keep on moving just so we can keep the show going. But thank you very much, Peter, for that presentation. And next we have Nick Indelicanto, who is from Mercer County, New Jersey, the supervisor of mosquito control. And he's going to present larval control of floodwater mosquitoes in Mercer County, New Jersey. All right, Nick, looks like I see your presentation. Just got to put it in presentation mode and you're ready to go.
Hello, everyone. Today, I'd like to spend my time with you discussing the larval control of floodwater mosquitoes in Mercer County, New Jersey. I'll begin by giving you a brief overview of our county, followed by a walkthrough of our annual calendar of events, which we use to plan and implement control measures to combat floodwater species of mosquitoes. I'll also share some of the most common occurrences we experience while performing this type of work, as well as our methods of intervention, and then wrap up with a brief discussion regarding our surveillance efforts. So jumping right in, you can see the state of New Jersey on your left and a map of Mercer County on the right. We're located in the west central portion of the state along the Delaware River within a suburban corridor that spans from New York through Philadelphia. The county of Mercer is approximately 226 square miles in size, with a population of roughly 380,000 residents who live within the 12 municipalities that we have. This map indicates all the areas of floodwater that we're responsible for each year. And because there are more than 30 of them, we prioritize each site using a color coding system, which I'll explain briefly. The red polygons indicate our priority 1 sites, while the green polygons indicate secondary sites. Those shaded in purple are number 3 on the list, and the smaller blue sites that you can see interspersed throughout this map would rank number 4. Zooming in just a little bit closer, this image is indicative of what we deal with predominantly in Mercer County as it relates to floodwater scenarios. And you can see this residential neighborhood basically surrounded by floodwater. And I think this perfectly illustrates the need for early intervention as well as a structured plan of maintenance that needs to occur throughout the year to provide continued relief to these residents for not only nuisance populations of mosquitoes, but those species which may pose a risk to public health as well. So now we'll start to go through our water management annual calendar, and that begins in the month of November. We start here because we've now had a chance to shift gears away from some of the requests for service from our residents, and have also had a chance to perform a comprehensive check of all of the sites throughout the county. Now we can prioritize the work, whether it's tasks such as clearing trails to gain full access to some of the more remote locations, or whether it's deciding on how best to complete the water management projects themselves. And by January, our water management operation is in full swing. We've found the greatest value in targeting flooded areas caused by beaver dams, and we attack this on multiple fronts. We bring in licensed trappers to remove beavers from these key areas, and then once that's accomplished, send in a crew to manually remove and deconstruct the beaver dam using hand tools. This helps to restore water flow and to reduce the flooding to levels that are manageable to our operations. I've also included some pictures along the right side of your screen that just show some additional examples of the type of damage beavers can inflict on an area. As we get into March and April, we begin to move away from water management and implement full-scale larval sampling. Each day, these specimens are brought back to the lab for identification to species, and during that time, we also determine their developmental life stage so we know when best to implement our initial larviciding program. This typically happens during mid-April, where we call for aerial larviciding from an outside contractor that we use. And you can see that pictured on the right side of your screen. One of our inspectors here is seen pouring a bag of granular larvicide into the hopper on the side of the aircraft. In this case, we're using Vector Prime FG, applying it at a rate of 4 pounds per acre. And we've seen encouraging results based on post-application larval surveillance. As we move through the latter half of April and into May, our larval sampling continues to ensure the success of our efforts, and then also we can perform some touch-up larviciding if we need to revisit sites that need some additional attention, or if we need to catch up to sites that we haven't had a chance to treat yet. And then if Mother Nature deals us a kind hand, the spring bloom tends to help dry down some of this floodwater, so we have less to deal with. Of course, if the opposite occurs, we're prepared for that as well, and we'll call in this outside contractor again to perform some additional aerial larviciding. As we get into June right through October, our inspectors are typically inundated with requests for service from our residents for a number of different reasons, including container inhabiting species, which is a whole nother presentation. But for these reasons, because it becomes so busy, we've designed these predetermined routes that exist within each municipality so that inspectors can fall back to those and continue to perform checks on these floodwater habitats throughout the summer. One thing I'll highlight here in the same vein as floodwater is the need to keep an eye on the functionality of detention basins, particularly in the spring and summer months when we have intermittent rain or torrential downpours that starts to overwhelm these basins, and then they become clogged and don't drain properly. And that can present quite a problem for us, which leads me into my topic, and that is the most common floodwater scenarios that we deal with. So starting from right to left here, on the right side of your screen is another basin that is malfunctioning. So some recent rainfall has occurred, and this basin can't drain because the trash racks are clogged up and won't allow the water to flow. And so if you think of the magnitude of the problem, I just want to put this in perspective for you. Because of the 12 municipalities that we have, several of them have hundreds of these basins within their municipal borders, all of which need to function properly. And so if they didn't, or if they were left unchecked, you could imagine the magnitude of the problem just looking at this one basin and thinking about how many mosquitoes could inhabit this one area of floodwater. And then next to that would definitely be the impoundments created by beaver dams. Because despite our best efforts, beavers are resilient and they tend to reestablish themselves quickly in the areas where we've just worked. And so they can rebuild right under our noses, and we're typically on a razor's edge trying to deal with that. And sometimes the situation becomes emergent where we have to drop what we're doing and treat these locations immediately. And then next to that would be these grass bottom pools, and these habitats are usually immediately adjacent to residential areas as well. And although you can't see any water here in this image, it's there, it's within all of those ruts and low-lying areas in small pockets. And there are several small pockets that in aggregate can produce a prolific number of mosquitoes that can really be bothersome and plague these nearby residential neighborhoods. So these are probably the three top contenders for floodwater scenarios that we deal with on an annual basis. So now I'll move into our methods of intervention, and most of this equipment that you see is probably familiar to most, if not all of you, but I'll go through them briefly starting in the upper left-hand corner. What you see pictured there is an amphibious vehicle. This is an Argo. It's an 8-wheeled vehicle equipped with tracks that allow it to maneuver through some of this marshy terrain that we often have to treat. We've outfitted this particular Argo with a broadcast spreader that enables us to dispense a granular larvicide throughout this habitat, and is an invaluable intermediate step between aerial larviciding and treating on foot, especially when time is of the essence and aerial applications aren't necessary. But again, treating on foot may take too long or be overburdensome to the operation. So moving over one to the right in the upper right-hand corner are the ATVs that we use. Typically, these are equipped with 35-gallon liquid larvicide tanks and electric pumps and sprayers to dispense a liquid larvicide in situations where vegetation has grown densely over standing water, and granular larvicide may not be as effective. Moving down to the lower right-hand corner of your screen is the helicopter, and I think at this point in the presentation goes without any explanation. But of course, if the weather doesn't cooperate with us and we have extensive flooding, we can call them in at any point throughout the season to perform large-scale aerial larviciding applications. But typically, what's most commonly used is one image over to the bottom center left of you, the bottom center of your screen. And this equipment includes backpack blowers like this Still model SR450, which can apply either liquid or granular larvicide. In addition to that, we have hand pump tanks, typically used for pupicide if it comes to that as a last resort. And then we have these smaller hand seeders, which can be used, just for touch-ups on smaller operations. And then in the bottom left-hand corner of your screen, I've saved one of my favorites for last, and that is simply fish. In this case, fathead minnows. We like to use these in impoundments or woodland pools that don't dry down throughout the season because they provide great control, again, season long, and it also augments our integrated mosquito management program and increases the positive outlook on our public perception as well, because we're using these biological control methods rather than solely depending on the use of pesticides to control mosquitoes. And then one of the last, but certainly not least, methods of intervention we have is communication. And whether that's directly with residents as we need to, or more importantly with local and local health and public works departments and homeowners associations. These entities can be of incredible value to this operation, specifically in regards to malfunctioning detention basins. And oftentimes, it's through no fault of their own, it just flies under the radar. And a lot of times, these folks aren't aware of the magnitude of the mosquito issues that can come from neglecting these basins. So once you reach out and explain that to them, not only have they been supportive to us, but they've provided additional resources to get in there and clean them up and maintain them so that we can focus our efforts elsewhere during the summer months when we're extremely busy. And that brings me to the heart of our operation, which is surveillance, surveillance, surveillance. Again, this is the backbone of our operation, and if I've said that once, I've said it, 1,000 times. This allows us to make informed decisions, no matter what time of year it is, whether it's larval surveillance in the beginning, mid, or end of season, or whether we're providing adult surveillance at some of these traps, which I'm sure are familiar to all of you. And going from left to right, we have a box gravid trap that targets Culex species, which allows us to send these specimens in for testing for West Nile virus and other pathogens, as well as the New Jersey light trap shown next over toward the right. We have 15 of these throughout the county at fixed locations that provide us invaluable data on what's going on with the mosquito populations and when we may need to perform some sort of intervention. And last but certainly not least is the CDC trap, with the CO2 component to it that we can deploy right near some of these floodwater habitats that give us a good comprehensive look at what species we're dealing with, so we know exactly how to target our applications. And again, you have to be able to provide the surveillance both before and after the applications so that you really know how efficacious your efforts were. And I think I'm going to leave that right there and just open up for questions. And I'll just end by saying thank you so much to Banu, to Leanne, to Jim Andrews, and to Valent for the opportunity to present before you today. My contact information is here. If you don't have time to ask me questions today, feel free to reach out anytime. But otherwise, I'm certainly ready to address any questions that you may have. Thank you all so very much.
Awesome. Thank you so much, Nick. I just checked the question panel, and we do have one question for you. All right. Are there any wildlife regulations you need to deal with when it comes to removing and relocating beavers?
That's a great question. Thank you for that. So we work with the New Jersey Division of Fish and Wildlife and licensed trappers to accomplish this. So whatever they are doing is already in compliance with the guidelines and rules and regulations set forth by that organization.
Okay. Awesome. I have another quick question. Do the fathead minnows dominate ecosystems like Gambusia?
As far as I know, not quite to that extent. And if I'm mistaken on that point, anyone in the audience is welcome to correct that. But they are more suited for woodland pools and that sort of thing, where Gambusia would be restricted to situations maybe like abandoned swimming pools or something like that, that we can, in a sense, guarantee that it won't flood over, and then introduce those fish into the ecosystem. So they're a little bit more friendly in that regard than Gambusia would be.
Okay, awesome. Let me just scroll here real quick. I think the rest of these are just prior to you, so we'll save those for save those for the panel. We do have one more question. Are fathead minnows native to the area?
Yes, I do believe so. Again, if I'm mistaken, anyone's welcome to correct me, but I believe they are, yes.
Okay, awesome. I think for now that covers most of the questions we have for you. There is a comment out there. Yes, the presentations will be available to you all after the event is over on the VBC webpage. So we'll make sure that we get that to you. It's also almost 12 o'clock, so I need to do a quick shout out that there is another pop-up quiz. It will be dropped in the chat for you to access that quiz. Again, those states are California, Utah, Colorado, and Oregon. Make sure that you fill out those pop-up quizzes. And I will mention one other time too, there is a text option. That link will also be in the chat. If you want to receive the pop-up quizzes via text, you can do that as well. All right. So we're moving right along and doing great here on time. So we'll go ahead and introduce the next speaker. All right, next we have Mark Smith, the Purchasing and Supply Chain Manager at Metropolitan Mosquito Control District in Minnesota. And Mark is going to present Applying Low Methoprene Rates by Aircraft. So Mark, if you're ready, you can share your screen and take it away.
All right. Am I up and running here?
We can see you.
All right, can you see my presentation?
Not yet, just you.
Okay, I'm sharing my screen.
Do you have two screens up? Maybe we're not sharing the right one.
And the share screen process actually has two buttons. First, you'll hit the share screen, and then it'll give you multiple options. Which one you want to share? You might want. Yeah, there you go. Something happened. Yeah.
You're up.
Okay, thank you very much. I appreciate the opportunity to participate in this event. Thank you very much. Starting off, I'm sorry. The Metropolitan Mosquito Control District is located in Minnesota, up in the upper Midwest, and our district services the Minneapolis-St. Paul area. We cover about 3,000 square miles in 7 counties and service a population of about 3 million people. Our annual budget is about $9 million, and we have 7 facilities in which we service our area. We have about 54 full-time staff and 210 people, seasonal inspectors in that. We contract out with 6 Jet Ranger helicopters that are for exclusive use for our program, and we focus on larval mosquito control. The MMCD has a long history of with methoprene. Our former director in the 1980s, Dr. Robert Trogan, worked closely with the Zoycon Corporation to develop a lot of the formulations, the Alticid formulations that are around today. And many of the use patterns and product evaluation techniques used today were created as MMCD became an early adopter of that those products. What we have here is a map of our 7-county metropolitan area. It is, you can see Minneapolis would be here and St. Paul there. You know, Minnesota is known as the land of 10,000 lakes. I think it should also include, we're also the land of 100,000 wetlands. So we have a lot of them in our district. You know, we have identified about 81,000 different breeding sites, which, you know, can be seen in red. And that habitat is included in, you know, both urban in the core of our cities, and then working out to suburban, and in the far reaches of our district, we have more rural areas. We do not have the resources to treat all these sites, so a big part of our job is to prioritize which ones will have the most impact on our citizens, you know, how we can control and do the best for the common good. And we try to utilize all active ingredients to operate in the most efficient, environmentally sensitive, and cost-efficient means possible. When we look at our operational strategies, we focus our control on areas of high population and surrounding areas in which would affect those areas. All our treatments are done according to thresholds and larval surveillance and site history. Our entomology lab will probably look at somewhere in the ballpark of 20,000 samples per year. And we utilize a database system, a GIS database system, which is our inspectors can directly access and input data and have real-time information using their smartphones. We utilize site histories quite a bit in this when we look at what sites to use pre-hatch or residual materials compared to like a shorter term BTI problem. And what we look at is if a site will produce mosquitoes like 3 times in a 30-day period, that would be a good candidate for using residual control. The majority of our sites are treated by helicopter. We usually target sites that are greater than 2 acres, with anything less than that being treated by ground. We're really all in on larviciding, since that about 93% of our control budget does go towards larvicides. And we usually target for evaluating control in our treatments as approximately about 10% of our sites. That may seem like a low number, but when you're looking at the thousands of sites that we treat on an annual basis, it does get to be significant. So one of the things that we focus on is, since we do all of our work with helicopters, is really doing a good job with that application platform. You know, it is really critical for these low rate applications, talking in the ballpark of 3, 4, 5 pounds an acre, that you do a really good job. And I mean, all of us know that if you get the product where the mosquitoes are at the right dosage, you'll have real good control. So that's really our target. One of the things that we did is we developed a mobile calibration system in which we purchased a 24-foot trailer and made a mobile lab, customized it for that. It's all self-contained. With the use of generator, we can go anywhere in our metropolitan area that we have a field large enough to set up and do calibrations or helicopter checks at that time. So what we end up doing is putting out a series of trampolines. You can see on the bottom of these collectors, the helicopter will fly over. In the lower left-hand corner, you can see each of these trampolines has a vial on it. We can collect the materials that fall out of in our swath patterns. We can bring them back into our trailer, measure the weight of that, and then eventually enter that into a statistical program in which we can identify our swath patterns. One thing that is important to know, each of these materials, no matter if you're looking at metal pellets, natural BTI, they all have their own unique characteristics, and each has its own bulk density, or aerodynamic qualities that you have to adjust for and look at. And when you're looking at these really low rates, those features really do come out, and it will show you many different patterns, and it's important that you understand the capabilities of each material and also know its limitations on how you can best utilize that and set that up for application. It's also important to note that each aircraft has, a unique application pattern. That when you're looking at, a lot of the rotary craft that we use, you have two hoppers on the side of your helicopter, and you need to be able to understand how both those units work in unison to produce your swath. Sometimes it's difficult to know what materials coming from which side, so a lot of what we've started to look at is using colored blanks in one side of that hopper, and that helps us understand exactly what's occurring in our swath pattern and how each hopper itself contributes to that different pattern. One thing also is to note that helicopter shape, the fuselage shape, or the amount of rotors that you have, there's 2-blade, 4-blade, 5-blade, and the different helicopters you have, that also can produce different patterns, and you might need to look at that fuselage shape or that rotor wash and that in helping you determine how to produce the best pattern for your larvicide operations. So once you get all this figured out, there's a point where you get to the operational side that you need to be able to get an operational swath distance. When a helicopter is putting out materials, you may end up putting out a 100-foot swath or a 90-foot swath, whatever, with that. But to be able to put out materials at a consistent rate, especially at these low rates, you need to make that cutoff where in the upper right corner here, if you look at, you may need to cut off some of those outliers in there and cut off to a certain distance in which will perform your swath in a consistent manner across all your sites. What we use is a statistical program to help us decide that. We use a program designed by a company called WRK, which was developed for the agricultural industry. They have a granular deposition software that helps determine what is the best swath for the material, and that you can look at the different swath distances, and it helps you calculate out the rates and some statistics in which can help you decide what is the best swath width for that particular material. For us, typically, we come out with an operational swath of about 80 feet for sand type granule, and then 72 feet for some of the lighter corncob materials. And that way you are able to, if you look in the lower right corner here, you can look at the projections from our AgNav tracking system, our flight paths, in which you can project that 80-foot swath on those, and you can help understand your treatments and look at and ensure that you have good coverage on all your treatments in that standpoint. You know, so how did we determine our application rates? You know, going back with that work from Dr. Sjogren and that, we did a lot of development work looking at what would be the best rate, you know, for floodwater control, and through trial and error, you know, we actually found that about 2.85 pounds per acre of Alticid pellets achieved about 90% emergence inhibition. You know, we used to have a program in there, about 8 people in our district here that just solely worked on efficacy and applications, and worked at a way to continually improve those. So we have a lot of extensive experience in evaluating, you know, these products and that. And what we kind of use is that pellet, that 90% EI, as kind of the standard in which we judge other programs or other formulations and products in the market, and that as we look at it, evaluating those. Operationally, we still see methoprene falling in, you know, the range between 75 and 95 emergence inhibition. You know, I will say that, you know, there's a lot of things that we understand about methoprene, but there's a lot of things that we don't. You know, we have, you know, from personal experience in that, I've looked at a lot of different sites where, you know, you might have sites side by side, treat them both equally, same time of day, same, you know, environmental conditions, and you'll get different control rates in there, and that you know, things that you can't necessarily explain. That happens sometime, but in the majority, you know, we get really good quality control from those products. How do we test? You know, we do a lot of laboratory and field bioassays, in which our inspectors would go out into the site. They would collect pupa, bring them back to the lab, and then hatch them out and be able to get percentage hatch from that. Of course, we use control sites and that to determine the natural mortality, and that is corrected in our testing results for the product. So we do a lot of that. And one thing to remember is that some things that might affect some of the rates in your bioassays is that when you are collecting pupa, you are removing them from the site and that source of methoprene. So when they're in the laboratory, they might not be receiving the same amount of methoprene as in the field, and that may have a small contribution to your efficacy results. But, you know, for the most part, we've had really good success using that method to evaluate our products. And we do use some emergence trapping, especially in some of our Clocotidia perturbans sites, where we can actually set up emergence traps, as you can see in the lower right corner. These traps are set up over the vegetation and that, and collect the insects as they hatch off. And there, I think we get a much truer reading of the product, because those insects, those mosquitoes, stay in that water with that methoprene source all the way up to that moment of hatching. So I think that's a good, solid evaluation method. And, you know, with our applications rates, economics always plays a part in our decisions. You know, we're trying to cover such a large area and maximize that coverage. So that does play a part in how we evaluate. So that gets a little bit in, you know, with pre-hatch control, just kind of go into a little bit of the, you know, philosophy is, you know, when we look at control with pre-hatch products, and that is just, you have to kind of make a decision on how much control is enough. You know, are you targeting 100%, 90%, 80%? You know, I think if you're in a smaller district, you certainly would want to try to get close to, you know, that 100% efficacy that you can. Sometimes when we're treating larger areas, there might be some trade-off to maybe get 85% 100% control in more sites as opposed to getting 100% control in fewer sites when you're covering such a large geographical area. One of the things that we like to look at is that when we look at all the tools in our toolbox and all the different control materials that we have, how can we have them work together to maximize our effectiveness? And we look at, I talked earlier about, looking at our sites that produce mosquitoes, 3 times in a 30-day period, that might be a way to maximize that effectiveness of your program. Other days when we're covering large areas, we might want to look at maybe, concentrating pre-hatch areas in certain hard to reach areas. And then when we do have the rains and floodwaters, we can concentrate and be more efficient in control with a product, let's say like BTI, in other areas. We're always trying to get better at what we do and get more efficient. So that is always a quality improvement is always a goal of ours. I think it's important. I know it's been spoken to on by a few of the other speakers that we need to take care of our AIs. We do have active ingredients. We do have a limited number of active ingredients, so it's important that you try to protect that use and avoid any sort of resistance, especially when you're looking at low rates. So I think you need a good management plan that includes product rotation in there to help keep these materials working at their peak efficiency. Some of the other things that we look at is duration of the products and that time frame. Do you have that residual control? When do you use it? We always look at it as a risk versus reward. Is the site going to be wet for that full 30-day period? Let's say later in the summer, that we may not risk putting in 30 days product if we're not going to possibly receive the 30 days of control. So we end up doing a lot of our control with the pre-hatch products earlier in the spring when we know those sites will maintain that wetness, or receive those repeated rains and that product will be fully reutilized in the spring. So some things to think about. For us, we have 2 main pest species. We have the Aedes floodwater species that are, one of our main pest areas, but we also have Clocotidia perturbans, which is another main pest species that come off. But in our area, the perturbans give us an advantage because they overwinter as larvae. We're able to survey those in the fall period and know what's going to most likely happen in the next spring. So, we look at weighing, we see that as a known, and we do some maybe heavily weight our pre-hatch program towards more of those perturban sites versus Aedes floodwaters, where some of that thing, if we have a dry year, holding back some of those control materials to use in those times, might be a gamble. So it's a really balancing act, and it's just some things to think about. Another area where we use a lot of pre-hatch is in limited access sites. We have areas like, let's say, around our international airport where they don't want helicopters flying in there quite a bit. We will use them there. Areas of high use parks where you have a lot of public interaction, or maybe horse farms, or around schools where you don't want that aircraft in, possibly creating some public relation issues, and that I don't think anybody likes to deal with those. So, and lastly, for us, we're all about efficiency and trying to get to as many sites as we possibly can in a given period of time. So, using these low rate materials, and that allows us to get more flight time with our helicopters. When our helicopters are flying, we want them to be applying materials, so they're able to take with these low rate pre-hatch materials, they're able to take more material and fly more acres, and not be returning back to ferry and gain more materials all the time. So we like that because we like to fly as many acres per day as that we can when we have good conditions to fly. Lastly, what I'm going to touch on is drone applications. I know some of the other people have used those. I think these low applications rates are ideal for their payload and flight time. We have done some work and had great success, and we are still determining what, how best to utilize or find that niche for that technology. Lastly, I'd like to say is that the Metropolitan Mosquito Control District does have what I think are some high quality reports and technical reviews that we do every year. Those may be a resource for you if you want to look at some of the work that we've done. And those are available on our website at mmcd.org. So with that, I am finished, and I thank you for your time today.
Awesome. Thank you so much, Mark. Stay close by because you do have a lot of questions in the Q&A. So we're going to go ahead and get through our last presentation, and then we'll come back to all those questions in the Q&A. All right. All right, great. So up next is actually our final presentation. And this is Banu Kesavaraju, the Global Technical Manager with Valent Biosciences. And he's going to present technical challenges and solutions for low rate applications. So Banu, when you're ready, you can go for it.
Good afternoon, everyone. So I'm going to be sharing some information about challenges we have for low rate applications. So first of all, why low rate applications? This on the label for everyone. So every year we go through this, choosing the right product for our needs. What is the correct label rate? What is the, how can you maximize the efficiency of the product that you're applying, keeping yourself safe, saving money, helping the environment? All of these things actually play a very big role in actually choosing the right product for you. One of the important things for the districts is actually lower operational costs. And what do I mean by that? So when you have a low rate application, you have a reduced ferry time. So if you have a regular aerial application, like in using an aircraft, so number of times that the aircraft has to go back and forth to refill actually is lower. Thereby you're saving on fuel costs and also on wear and tear on the equipment. And if you're actually contracting, then the time that the aircraft is up there in spending for you for doing all the applications. But as Scott's talk was there before, and quite a bit of interest that we got from the questions that came after his talk, the topic of the right time now is the drone. So how do you use drones to actually do applications? So we have several spreaders that are out there in the market now for using drones, but drones have a limited capacity for us right now with respect to timing. So how long can a battery last for a drone to do the applications? So those kind of scenarios when the low rate applications actually make a lot of sense. So you can have efficient battery usage and also a longer application time with the drones that you have, if you have a low rate application product. Okay, so characterizations, we do this. This is the traditional way how we set up characterization. So you put some buckets or totes, containers you see on the picture here, and then you let the aircraft fly on top of it, and then you collect the materials back and put them in the cups, and you evaluate the weight of those granules, and then that's how you do characterizations. Now, some of the important variables that go with these characterizations. Coefficient of variation. I'm sure many of you would have heard about coefficient of variation, but that isn't actually an important variable when you're actually doing these larval granular characterizations. What does it measure? It actually measures the extent of variability in your application. Well, there's a lot of statistical jargon to it, but it's on Wikipedia. So if you search coefficient of variation, you'll get that. But in a nutshell, what it is that you might get a mean or an average for whatever you're doing. But that average, what does it actually represent the whole population, or does it represent only a small portion of the population? So let's take one scenario here. So here in this graph, you can see from A to H, these applications had different trends. So the bar graph actually it keeps increasing. And in the end, you see peaks on there. So a coefficient of variation here, this is not actually, if you get an average of this particular sampling, you won't be able to get the right average. The average is not actually representing what's happening in the field. So in this kind of a scenario, you will have a COV, which is actually high. So anything at the range of 1 is pretty high coefficient of variation, just saying that the data is too variable. The mean that you have is not really the mean that you can actually use for deciphering anything, because it's actually have these infrequent distributions. But let's consider another scenario. In this scenario, you have even distribution. If you look at the COV for this one, it is pretty small. It's only 0.03. And this is actually, if you get a mean from this distribution, then your mean or average is actually more reliable. So whatever you're trying to achieve is more reliable. So in our recommendation, we always stick with the COV of 0.3. Less than 0.3 is better when we're doing these characterizations. And another important point with these applications is the minimum preferred application rate. How much should we apply? At any one point in the swath, how much is the minimum that you need to have? That varies by habitat and species and the product that you choose. But each product will have a minimum application rate below which you do not want to apply at any one point of the swath. Okay, so what is an uneven distribution? We see this very much in lower applications issues. I mean, we do see this when the equipment is not calibrated and configured properly, that these uneven distributions, even in a high rate. But typically, it does occur more often in the low rate applications, hence this focus, and we're trying to look at it. So in this graph, you have pounds per acre, and these are data from the tubs that have been placed across the swath here on the x-axis. And these are actually the pounds per acre applied. And you see this line, the red line, that's the minimum preferred application rate. That is 2 pounds per acre for this particular product. And your targeted application rate, which is the green dashed line, that's where you want it to be. That's your ideal. That's what you're actually targeting. Look at the average here. That's 2.6 pounds per acre. Can you trust that average? Look at this peak here. At one point of time in the swath, you're getting 10 pounds per acre. Some about 9.8 pounds per acre. So there's quite a bit. So if you actually get this kind of a distribution, then you have an uneven distribution. Not only that, that's over applications. You're wasting product because all of that is going to be staying in one area of the swath. Although your targeted swath is 55 feet, and that's what you are going to be using, but your actual swath is only 15 feet. And that's because this is actually streaking, and you have a problem with the equipment, and it is actually applying in one area, and it is not distributing evenly. Okay, let's see some low-rate application examples. How can we actually resolve them? So I picked Vector Prime because it's easier. I'm more familiar with the Vector Prime, but this applies to any other low-rate application product. You can choose anything that's out there. And for this, actually, according to the label, for post-flood situations, if it is not polluted, it's about 1.25 to 4 pounds per acre. That's a really low rate. There's almost half of some of the traditional larvicides that are out there. So achieving this, what are some of the challenges that we might actually come across, and how best to resolve them? Up front, collection containers. When you're doing these characterizations, before even you go into equipment problems, first things that we have seen in the field is actually what kind of collection containers do you use? So this is a typical, as you see in the left picture, where you actually line up all the containers, and you have an aircraft that is actually flying on top of it, and you collect the deposits, and then you look at how many pounds per acre at each one point of the swath the application was. Now, in this case, what we did was we did a comparison between tote boxes, which have typically a little bit of a bigger opening, and then a bucket, which is actually a smaller opening. So is there any difference? Traditionally, a lot of people use buckets. We've used buckets a lot. But what is the difference? How do the tote boxes actually perform? And all other things here are similar. So same aircraft, same opening, same product, wind speed, temperature, everything is same because you see both of them are lined up exactly. Is there a difference when you do these kind of applications, especially when you have a low rate product that you're trying to evaluate, characterize your equipment for? Let's look at the results. So left graph is for tote boxes and the right graph is for buckets. Again, everything is same. The only difference is whether they were actually buckets or totes. But other than that, all other factors are similar. And you see on the left one how the distribution is a little bit more even for the product. But if you look at the bucket on the right side, there seems to be some peak. Again, it's same equipment. Everything is same. How can this be? It looks like actually the amount of opening, that the mouth of the collection container that you keep actually has a lot of impact on these low rate applications. This is more profound in low rate applications than in high rate applications. So this is something that we have to keep in mind when you're characterizing some of the equipment, is to make sure that you have a container that actually represents a little bit of a bigger opening than a typical bucket that's a smaller and circular. And if you look at even the COB here, that is actually 0.3, it's pretty good. That's what you would recommend for an equipment for a low rate application. So this one says this is qualified, but on the right side with the bucket, this says it's 0.7. That means no, that run was not qualified. That equipment is not actually doing what it's supposed to do. So that there itself, you have a confusing information. So you got to choose the right method to put it there to collect your granules in order to do the characterizations to see if you're able to configure your equipment properly. Okay, moving on to the next one. So this is actually a predicted swath pattern. So once you actually have a typical pattern, you put it in a software or some Excel sheets where you actually do these simulations to see how well, if you do an aircraft back and forth pattern, how will your deposits be? Again, on the left side, you have tote. On the right side, you have the bucket. Everything else is the same. And you can see the streaking that you're actually getting in the bucket. That's misinformation just because you had the wrong type that you put under the aircraft to actually collect your containers. But on the left side with the tote, it says everything is good. And when you extrapolate this for 360 feet of back and forth, you're doing perfectly fine, no problems. So good one to keep in mind when you're actually characterizing these equipment. Okay, I'm going to go through a couple of case scenarios pretty quickly here. This one is actually a fixed wing aircraft. There was an issue where there was streaking. So what is the best way to resolve it? Actually, installing a custom roller actually helps a lot. So that actually improves the swath. The rollers are available for different kinds of equipment. Even Isolar actually has a roller now. So you can use those to actually equally get the product out. And this helps especially in low rate applications. There are rollers that can actually handle smaller application rates very efficiently than higher application rates. So roller is a good solution if you want to go with the roller method for applications. So for this one, it was 4 pounds per acre target and then optimal swath with this 90 feet. So we'll go through a couple of graphs here. You're familiar with the graphs like just before. Good even distribution, and then COB was only 0.25, which is good. The equipment is doing good and the calibration is pretty good. On the right side, the predicted swath also indicates the same data. So that one is good. Let's go into case 2. This one actually is a rotary wing aircraft. Again, it shows uneven application. What is the best method to resolve this? And in this time, in this equipment, in the picture you see there's a tube that is coming out. There's actually a boom that's actually attached to that tube. And the boom actually has 4 points where it actually discharges the granule. And this is another way of actually evenly distributing your low rate granule. And you have smaller hopper openings, testing those different types of hopper openings, and then the boom that was there actually helped in alleviating the problem, so you have more even distribution across the path. For this one, was 5 pounds per acre, and the optimal swath was 75 feet. There's a couple of graphs here, and if you see on the left, again, even distribution, no issues. COV was 0.3, which is good, and that's what you want it to be. On the right side, if you see the simulation, the graphs look really good for application. All right, so moving on. Some of the key considerations. So what would you do? First and foremost, for characterizing the equipment, you want to use the right type of container that you're going to place under those aircrafts or drones for that matter. So totes are a little better, mainly because of the opening that is there. So the bigger the opening, better you're going to be in estimating closer to your true value of the true average of what you're actually getting. Low rate applications are going to be the future, especially with drones, and we have limitations on all of how much it can carry efficiently, and also increasing the payload so that you're able to cover more area. So moving towards that, we need to pay attention to all of these important features so low rate applications can be effectively applied. Existing equipment can be modified and configured accordingly. It's not a big deal. You just have to be focused on the characterization to identify the problem first. And when you identify the problem first, then you'll be able to resolve them pretty quickly, regardless of whatever equipment you have.
All right, thank you so much, Banu, for that presentation. We do have quite a few questions to get to, so we're just going to go ahead and go into the panel discussion. So if all of our presenters will kind of come back, turn your cameras on, I'm going to kind of go through these questions. I may have to go kind of in order just so I can timestamp these to know who they go to. So Banu, we're going to go backwards and just start with you since you just finished. Is there a recommended surface area with the totes to best characterize the low rate dosage?
For the tote boxes, the ones that you saw in that picture and the ones that we have constantly used is the one that you get from Walmart. We bought them for those 18-gallon tote boxes. Those work fine. I think they have about 28 inches of the width and stuff. So, but you can use that as a reference and go from there. But yeah, you can test different options. The 18-gallon tote boxes at Walmart is perfect for us.
All right, awesome. Still for you, when doing low rate applications, does that reduce the chance of the target pest becoming immune to the product?
Now, so just because it's a low rate application does not mean that you're actually under applying below the label. That is the important thing to keep in mind. If you're actually applying below what the label is suggesting, that's where issues crop up when you are actually doing so resistance and other stuff because you're exposing them. And also the type of the product also matters. So if it is BTI, then that's not an issue. So it depends on the product that you're applying, and most definitely what I'm suggesting is low rate application by the label. I'm not saying just low rate applied besides the label. So if the label says so, then you're not actually applying below that you're going to be selecting something.
Awesome, thank you. We're going to move on to Mark Smith. We have one here. Were those trampolines that you use for swath characterization made by you folks in-house, or are they commercially available product that you can purchase or maybe modify?
Well, we did have, when we purchased the system in 1993, they did a similar trampoline was included in that. We have since gone through multiple versions of that, same sizing and everything like that, but they do wear out over time. They should be available through WRK in some form. But yeah, for the most part, we have kind of had them redone by a private fabric manufacturer and that to rebuild them for us. So
great, thanks. Got another one here for you. What is the percentage of active ingredient of I think it's Alticid for applications at 2.5 pounds an acre, which received 90% emergence inhibition?
Well, the amount of methoprene in the product for Alticid pellets is 4.25%. I don't have the calculations. If she's looking for some more detailed information on the actual amount, I don't know exactly what she's looking for there, but the label amount is 4.25%.
All right, great. Thanks. I'm going to keep on moving here. I'm kind of using my timestamp to see who these belong to. So Mark, I think you're still on. Do you have thresholds set for adult mosquito control intervention in the case of disease outbreak? If so, have you ever exceeded them with your extensive larval control problem?
Well, fortunately, we don't have a lot of disease or vector activity in our area. We do have some eastern equine and some Jamestown Canyon, but I guess if we looked at a disease outbreak, we definitely look at our thresholds, and we do target our Culex species, which are our vectors in our area, and that they do have a little lower threshold for treatment in our area. So when we do find some of those Culex species in that, we do address them accordingly. So.
Okay. Awesome. I got definitely one more directed right at you. How do you rotate your 10% of sites to evaluate larval control?
Well, I guess to look at that, the same sites don't always get wet in every rain. We cover such a large area, so we're constantly looking at different sites all the time by which region of our 7 counties got wet. Our staff will try to target of the treatments that were made, 10% of those. So it isn't just a continual same site evaluation type of process. It's kind of evolving and rolling as weather conditions and that roll through our Twin Cities area.
Okay. We have a question about your colored blanks. Did you guys make those colors yourself, or did someone provide them to you?
We have asked some of the manufacturers in that to provide those to us. Sometimes that's not always easy to do, create a blank colored product in their manufacturing processes and that. So some of the manufacturers have provided them. Some we've done ourselves. We use a phosphorescent dye on some of them and mix that just prior to putting them in the hoppers and that to be able to distinguish those 2 hoppers. And one thing with that phosphorescent dye, it's pretty cool because you'll put it into one of the hoppers and you'll have like a pink streak going across the sky as they're applying them, and that's a little bit of an air show there too.
Awesome. All right. For the moment, you're off the hook, but we got Nick, I think a couple directed at you. We have a question that came in right when you were talking about your surveillance tools. And we have a question from Virginia that asks if those devices are legal in Virginia. I'm not sure if you'll know that, but we'll pose it to you anyway.
If you're referring to the mosquito traps, I couldn't speak to the legal status of those traps in Virginia. I certainly haven't heard that they're illegal, but wouldn't advise any legal advice one way or the other there. I can only speak to our county operations in New Jersey.
All right, great. This is coming in your time frame, so I'm going to go ahead and read it. But if it is for someone else, help me out here. I'm pretty sure. Okay. I'm pretty sure it helps you all combat a large amount of adults, especially since you are quite limited about the areas that can be treated. What products do you use? And we switch a lot from oils to water depending on specific species. So what kind of products do you use in some of those limited areas?
So speaking to Mercer County, we use a variety of different products, mostly water dispersible biopesticides, your BTI, your Sphericus. We'll use methoprene, we'll use spinosad where we can. And I think most importantly there, I would just highlight that we like to rotate those products, of course, in the interest of insecticide resistance. And as far as like monomolecular films or surface oils, that's only as needed if we have an emergent situation. Typically, we address those things before that occurs.
Okay. Awesome. I think I have one for Mike. It's about your time frame. When spraying 12AS from the 206, what is your application rate of mix?
Oh, it's 32 ounces an acre straight. Not getting diluted.
All right. That was definitely, you knew that one. All right, let's see what else, if we have another one. Do we have Dr. Tiemann on the line? Nope. Okay, we'll
No.
say.
No, I'm here.
Oh, yeah.
Awesome. So we do have a few questions for you, if you're ready.
I am.
Awesome. All right. Have you observed any resistance in Sphericus in tarsalis?
So my lab has only done work with pyrethroids. We've only looked at adulticides. So I don't have the answer to that.
I'll pick up the question there, Leanne.
Please.
So far there's no reports out there for tarsalis and sphericus resistance out there. A good person to contact, of course, would be Stephen Ceu. He's done a lot of work with the BTI and B sphericus resistance and all other types of resistance. But so far I have not seen any publications specifically for tarsalis and sphericus resistance.
They also asked the same question about restruans.
Same thing, restruans also not common. Restruans are more often in the containers, and sphericus people don't often put them in containers, mainly because Aedes mosquitoes are not very susceptible to sphericus. You need to put a large amount for sphericus to be effective against Aedes mosquitoes. And for that reason, they don't come into contact with that very often. So far, there are no reports specifically for restruans that I know of.
All right, great. We have another one here for you. It is, where are your susceptible populations from? And what generation are they if they're lab reared?
Yeah, so both colonies that I use. So I know my talk was recorded this morning, or was a recording from this morning, but depending on the day, we use different colonies because tarsalis colonies are infamously difficult to rear. And so finding mosquitoes to use on any given day is pretty tough. So we had different, they came from different places. Like we had some that came from UC Davis, we had some came from local vector control districts, et cetera. Historically, where they started, the BFS colony, that stands for Bakersfield Field Station. It's a colony that was started, I'm not sure I could give you a year, but decades ago, collected from Central California, from the Bakersfield area. It's been maintained by the CDC and others for many generations. Same for the KNWR, that stands for Kern National Wildlife Refuge. It's a separate colony, but also collected from that Central California Bakersfield area. So they have been in colony for quite some time. I do not know the exact generation number, but it's been a while. We actually tried this last year to find susceptible for the last 3 years, give or take, to find susceptible wild populations to try to start a new colony. And we've had so much resistance in our area, we haven't had any luck. So that's kind of where we stand at the moment.
Another shout out if you have any, she's looking, so send them her way.
I'm looking, though. You'll have to be on the West Coast or in the Western US to help me.
Okay. All right. So one other question. Theoretically, would an insect population be expected to develop resistance more quickly when using a pyrethroid product without a PBO than with the PBO?
So the PBO helps combat the enzymatic resistance, right? So I'm trying to think if you're maybe specifically talking about KDR mutations, if it would affect that or not. The idea would be that the PBO would keep the mosquitoes' oxidases from breaking down the pyrethroid. In that way, it may lower the dose that the mosquito's getting. I don't know exactly what to say in terms of, so the PBO directly combats the enzymatic resistance. I'm not sure how to answer that in terms of whether it would affect KDR coming into a population. So the deal with target site mutations is that they're random mutations that happen, but then they have this evolutionary advantage, right? That those individuals that have that random DNA mutation survive, so then they pass that mutation on. So it wouldn't necessarily keep the mutations from popping up in the population.
Okay. Awesome. I'm going to have one more specific for you, and then we'll go and move on here a little bit. When tarsalis are exposed to the permethrin plus PBO, was it first exposed to a bottle coated with only PBO and then exposed to the bottle coated with permethrin, or was it a PBO mix, permethrin mix?
Yeah, so that's a great question. I know that, or at least I think still CDC guidelines say to do the PBO exposure first and then go into the permethrin bottle. That's actually not what we did for a couple of reasons. First of all, we were limited in time, right? So those of you who have done bottle bioassays know they already take a long period of time. So the pre-exposure we weren't able to do, but we also came to the conclusion to put them both in the bottle because that, to the districts that I was working with, seemed like a more realistic way that the products actually sprayed. So we didn't do them as separate exposures. The PBO and the permethrin were in the bottle together.
Great, thank you for that. All right, Mark Clifton, we're gonna send one your way. How much labor savings compared to backpack treatments using the Buffalo turbine? Are you treating 6 acres a minute with the Buffalo turbine?
Yeah, we could treat 6 acres in a minute, no problem. And that would take, hours or even a day with a team of 2 people with a backpack, depending on how thick the brush is. So we've found a dramatic savings. Some places that would take 2 days to get through, we can do in a morning. Depends on the wind. The wind has to be going the right direction. Our area is crisscrossed by lots of roads and paved paths. So we're lucky in that regard that we can use the wind to our advantage. Not everywhere would be able to do that, especially in more rural places. It might be more difficult.
Awesome, thank you for that. Mark Smith, we have one directed to you. Are you treating your Cochleatidia in the spring or in the fall? And if in the spring, have you ever tried the fall?
We've been doing Cochleatidia perturbans treatments in the fall for probably about 6 years now. We use Vectolex product in the fall. We do a lot of surveying early in the year so we can get some of those treatments done. Being up in Minnesota and that, we do, our temperatures start to drop after a while, and we target our Cochleatidia in the fall before the water temperatures turn 50 degrees or lower. We think that the activity of those insects really starts to drop off as the temperatures cool down, and our success rate for a product that they have to ingest like Vectolex is that we want to get it in the water when they're still really actively feeding and trying to build those stores for winter. We also do treatments for perturbans in the spring. We use Vectolex in the fall, and we use a methoprene in the springtime. We'll try to get all those treatments done in late May, early June, so we can have that coverage of that 30-day duration of that product throughout their whole time that they are emerging.
Great, thank you for that. All right, Dr. Tiemann, we're going to pull you in here again. Would you expect mosquito resistance to pyrethroids like permethrin to also be resistant to natural pyrethrum?
So this isn't an 100% answer because I haven't tested it, but yes, particularly for the KDR mutation, I would expect a similar effect, whether it's a synthesized pyrethroid or a natural pyrethrin. And then it's my understanding from vector control partners that often if you're spraying a natural pyrethrin, you can't use the PBO, right? If it needs to be in an organic field or something like that. So that also then takes away sort of that synergist effect of the PBO.
Awesome. Mike, we have one here for you. It's kind of a follow-up, similar to the first one. But for your 12AS, what was the application rate? You did say neat. And what was the droplet size, if you know?
Oh, the droplet size I am not sure on, but again, it was 32 ounces an acre, undiluted.
All right, great. We have a question posed to all of the presenters, so we'll kind of go around and throw it at all of you. Do you know any researchers who work on biology and ecology of spring woodland 80 species, such as vexins or Canadinus? Sorry about that one. Anybody out there familiar with that? I see heads shaking, but nobody's chiming in. All right. Yes, it's hard to get details on these species, and that's why everybody's shaking their head no. So sorry we couldn't be more helpful, but if anybody has that information, let us know. All right, let's see what else we have here. I'm just going to go maybe 2 more minutes. We still have a lot of questions. Mark Smith, we have one directed at you. What is the program that you use for your field inspectors to add data and information in the field?
Well, it's a program that we actually created ourselves, and that we were kind of on the forefront of, the database management when a lot of these programs came in, and it got to a point where it was hard to fit a commercially available program into what we already had established. So Dr. Nancy Reed at our office, and that does a lot of our database work. She works with a company called Houston Engineering to help develop our programs and that, and we also try to utilize a lot of open source type products and that in wherever we can to for our database work there.
Awesome. I'm going to aim this one towards Peter Brake and anybody with drones, but this is a drone question about what type of canopy penetration are you experiencing with your drone applications, with granular larvicide, that is?
Well, we have seen pretty good penetration with the material through the canopy. We haven't gone back into the field and have physically checked the material on all of our blocks, but we do go back every now and then to verify both with pre and post dip. So we get good penetration with our material from drones.
Any other drone people out there want to jump in? All right. Well, there are still quite a few questions, but unfortunately I am running out of time and I want to make sure that I have a moment to close out. So if we didn't get to your question, we apologize. We will continue to kind of work through them. Feel free to send them to any of the presenters. Like I mentioned a couple times before, their presentations will be available on our webpage after the event. Many of them have their contact information. So if there's a question that we didn't get to that you really want to know the answer to, please reach out to them or to one of us and we will get you that information as quickly as we can. Okay, I'm going to quickly go through a few closing things here just so we can wrap our day up. So I'm going to share my screen real quick to keep me on track. All right, can you guys see what we have? All right, well, we made it to the end. First of all, I want to take a moment for a special thanks to our speakers. Thank you for the time that it takes to prepare your information and present your information, and in general for everything that you guys do to protect human health with vector control. You guys are out there leading the way and we're all learning from you. So thank you so much for just taking the time and sharing your experiences and your knowledge. This is really the best way that we grow. And thank you for all the participants who are out there who watched the whole entire event and actively answered questions and asked questions. We really appreciate your time as well. I do want to go over a few quick CEU things. If you haven't already received it, we are dropping this Claim Your CEU forms in an email right to you right now. Remember, on that form, you follow the link, you choose your state and select the state name, and it will take you to that form for your state. Remember, state of Virginia, you do have an extra presentation in there to look at, so give yourself some extra time. And of course, if you need extra assistance with any of these forms or anything else, please reach out to us. There is one more key thing that you're probably all waiting for, which I should probably share with you, and that is the program code word. And the code word is DROP. D-R-O-P. So on your applications for CEUs, your claim forms, this will be the word that you enter for the code word. All right. Again, thank you all for your time today and your participation. Again, I know things are still difficult out there and we haven't been able to come out and see you face to face, but remember, the VBC team is here always to help you. We're set up to do virtual technical assistance, and whatever we can do to help you, please reach out to us. Have a great day, and we'll all see you later.
Thank you.